a very large faculty, a very large group of experts with us today. And I really hope that you will find this workshop useful. We hope that by the end of this few hours, you will have a better understanding of what are the NGS, the spectrum of the NGS technologies and the possible application, as well as you understand a bit better the analytical basis of the mutation catalog. You know, what is behind the standardization that we have recently published. I think I will stop here, and I suggest we move straight into the webinar, the discussion. I will introduce only the first speaker, then the speakers will introduce themselves. And again, we want to keep this as informal as possible. So please type your questions in the chat, or keep them for the Q&A session. The first speaker is Dr. Anita Sures. She is a senior scientist at FIND and the head of sequencing. I don't know if Anita is online. Yes, and she will talk about the role of sequencing in the diagnosis of drug-resistant TB. Over to you, Anita. Thank you.
Thank you, Matteo. I hope everyone can hear me fine. Good morning, good afternoon, and good evening, everyone. Really nice to be here, and thank you to the organizers and Union and WHO for having us and for this opportunity to share with you on this really exciting workshop. So I think today my goal is to be able to at least set the stage for all the more detailed discussion that go on, and really give an overview also of all of the other work and contextualize the work that's being done for this mutations catalog. Matteo gave a nice link and overview of the importance of this. I will place also in the context of everything else that's needed for drug-resistant TB diagnosis. So I have no conflict of interest to report here. Now, sequencing for drug-resistant TB diagnosis, again, I guess I'm preaching to the choir here. Most of you are very familiar, this is your space, but I think sequencing really presents a paradigm shift. And why we say that, because we know universal DST is still not there to end TB, it's still not been achieved. We also know there's less than 40% of DRTB patients are getting diagnosed, only 1 in 3 are getting treatment, and that's still the case with the latest report, even though there's a lot of achievements, a lot of good progress that's been made. And of course, with COVID, there's been a lot of setback in those timelines as well. So next-gen sequencing for drug-resistant TB really, as I said, presents that different approach in a sense of there's a lot of advantages it can offer. So one being it's rapid, definitely, compared to phenotypic DST. Just illustrate here in terms of the rough timeline for starting from culture for phenotypic DST. This is in the case of liquid culture. And then within sequencing, when I say rapid, there's of course whole genome sequencing, which starts from culture today, or targeted sequencing, which can really go direct from the patient sample like sputum. The second advantage, of course, being it's comprehensive compared to probe-based, PCR-based approaches. A lot more coverage, as many targets as you ever wanted, you can cover in that assay. It's also adaptable to new genes and mutations over time. So as you uncover more and more mutations, more and more targets, these can very easily be added onto the assay without having to go back to the drawing board to start R&D again or fresh to the development. Also, it's naturally high throughput, so it's scalable to whatever your needs are. So really targeted sequencing in particular could really revolutionize management of drug-resistant TB, allowing for faster, safer, and more comprehensive results to be able to direct clinical decision-making. But why is it then still not being used already today, you might ask? Well, there's a few challenges that are still there. So one is, I think, the whole being able to do direct from clinical sample, of course. It is still a signal to noise issue because you're starting from your sample, being able to take a direct, in this case, a sputum sample, you're going to have a lot of the human cells, you're going to have other bacterial flora in there, and you're going to have, if there's TB, some of the TB cells as well. And going through the typical workflow, you're collecting the sample, extracting DNA, processing, and sequencing. What you're getting is going to be then sequencing all of that. And that makes it, that's a problem. You're going to get a majority of human sequence and a lot of other noise, basically crowding out your MTB sequence that you're really looking for. There's, of course, a lot of technology ways to overcome this, to enrich that DNA signal from TB. So some of them, if you still want to use whole genome to be able to get the full coverage of the genome, there are options that groups here have used as well, with early midget culture, early liquid culture, in a sense, to be able to take that in advance for a 5 days in sometimes, to be able to use that as a starting. So you're still getting enough of it enriched in a sense of the MTB culture, but then still being able to do the workflow as is for the sequencing. There's other methods like RNA baiting and differential lysis that have shown. So basically you're taking along the workflow, you're going at different steps and being able to enrich for MTB, be it at the DNA level, be it at the lysis level itself, be it at baiting the appropriate targets, and so on. But these are all, of course, these are still not optimized. They're not in a state to be able to routinely deploy in a standardized manner around the world. So today, what is a good solution to really tackle this is using targeted sequencing. So really, which means a step of multiplex PCR in between, which adds, of course, the requisite few hours as needed, 1 to 3 hours, which allows you to really hone in on the targets, the regions. So it could be large genes, it could be regions of the genome, it could be multiple targets, as many as you want again, but you still have the multiplex PCR step, which then allows you to go to sequencing and really sequence and get only the TB sequence of your interest. This is greatly, as I said, that has been a big advantage of targeted sequencing, because you want to be able to do it if you're directing patient treatment, to be able to really go from clinical sample. Sorry, could people go on mute, please? So for whole genome sequencing versus targeted, we already went through some of the differences. There's obviously advantages to both, depending on what you can do and for what purpose. Since my purpose of this talk is really on DSD, for diagnosis, really the advantage with your whole genome, of course, it's comprehensive, you're covering the entire genome. It's definitely a step up faster than phenotypic DSD, and you're still getting that comprehensive coverage. There's a variety of applications, not just for diagnostic, obviously for surveillance, other applications. It makes a lot of sense to be able to get as much of the coverage, as much of the genome as you can. The limitations today is that you still need culture. There are groups that have looked at methods where you are able to sequence quite well starting from a direct sample with some processing, but this is still, again, as I said, not yet standardized and optimized for routine use. And also, in general, you're getting a lot more data, so there's a lot more complexity and cost in terms of managing that data, just the storage management, analysis, all of that. So all of those, and you come to targeted, becomes an advantage. So now you're only looking at, while you're looking at only specific targets, so you might miss out on things that you don't know yet, that you're not aware of yet, but at least you know all the targets that you can sequence at one time. You're enriching for regions of your interest, or different organisms, depending on what you're sequencing. The speed is there, and also the sequencing data analysis then is really linked to your target. So that makes it also, in terms of size of the sequence sample and so on, the data also becomes more manageable. So I'd find, or really from this project, our focus has really been on an end-to-end targeted sequencing solution. So being able to start from sample, go to DNA extraction, the library prep part, and then the sequencing, followed by the the data analysis and interpretation, which is then really directed by the interpretation is directed by the mutations catalog. So I'll come to sort of this different bits of this as we talk, but really I wanted to emphasize that the specs for this end-to-end solution, if you will, is really derived from the target product profile that was published in the WHO Technical Guide of 2018 for sequencing. And then the sort of interpretation for this is really from sort of the mutations catalog, really driven by a WHO knowledge base. So to make sure that as much as whichever companies, whatever solutions they have, whatever their targets they're reporting on, there is a list of truth or a reference standard that they can basically have as their lookup file to be able to report on all of that. As long as their assays cover it, which they should as a targeted sequencing assay, they can report those. And these were the targets, so really based on that target product profile, but you can imagine having a standardized set and getting the evidence, clinical evidence for this will allow us to really then be able to expand targets and so on much easier after this first step. So a little bit on this project, sort of the basis of this whole, the mutations catalog work and the targeted sequencing work is really this Unitate-funded Seek and Treat project. It's really focused on improving drug-resistant TB diagnosis. And it's really designed to generate evidence and also catalyze capacity to support global adoption of these sequencing solutions to be able to do comprehensive diagnosis for TB. And the 4 different outputs really focus. The first one, which I'll cover a little bit, would be on the clinical evidence dossier. And this is to get enough of the evidence to be able to present it for WHO review guidelines committee or technical experts and so on, to be able to get policy that can guide the use of targeted sequencing for DRTB. And the idea with getting global policy that can hopefully eventually also drive or shape or allow to inform national policies, regional policies, and so on for the use. The second key output, which obviously hand in hand with output 1, is to make sure, okay, so we have the lab workflow figured out here, but what does the lab workflow, what do the results really mean? So having a mutations catalog and knowledge base that really gets that standardized sort of set of mutations, set of targets, and the standardized analysis interpretation, that's another key to make any sequencing, in a way, a sequencing project or a sequencing workflow really make sense and be deployable. So that's another key output here. And with sequencing, while it's not just a one-box solution, because you still have a workflow, you still need to go a little bit further to be able to do some amount of implementation guidance, or at least some considerations for deployment in a reference lab in different settings for different purposes. How does a country or how does a program deploy this in the real time? How does this compare to standard of care? What are the operational characteristics and the advantages or disadvantages versus what they're doing today? So that's another part of this project as well, and really taking it all together to prepare for scale. So one big aspect of this is to ensure these products are listed on a global procurement listing so that countries are able to procure them today. COVID has obviously accelerated some of this, because some of these I think all of the sequences actually that are used for this are also being used for COVID, and hence they are actually already listed on the Global Fund catalog. So there is some of the sort of advantages of sequencing already being used so widely we are seeing today, which I think the TB programs in a way should really take advantage of. And the output 1, as I mentioned, which was really on the evidence generation, just want to kind of talk through what we have done so far. So what we have accomplished or completed has really been the analytical validation in the last year, really towards the early part of this year as well, which is really with the blinded reference panel completed by 3 different manufacturers of end-to-end, bringing together 3 end-to-end solutions. So based on the analysis, based on review from WHO, Unitate, and a technical expert committee, it's clear that they meet the criteria, the initial criteria for a phase 1 analytical validation, and they're suitable for phase 2. I'm not mentioning the manufacturers here yet because we're pending contracts with each of them, but happy to talk offline if you all have specific questions. But you can imagine there's not that many manufacturers of DRTB sequencing-based solutions today, unfortunately. But let's hope everyone who is in it, and there's more people that want to be building this, that would be great for the community. And the clinical evaluation piece, which is really the next piece. So we've seen it on an analytical lab validation that all the mutations that the assays are supposed to detect, they're able to detect. And now going to the clinic to make sure in a clinical setting, the controlled setting, they're still able to perform as is with real clinical samples, with prospective samples. And so that's where we are now, where we have been doing the patient enrollment in 3 countries, and also reference testing and so on that have already started. And the idea is to be able to synthesize the data from the TNGS solutions over time over the next year to be able to get it ready for WHO review. A quick one on the analytical findings, which I think we've also presented at another session, so you might already be familiar. Apologies for those for whom this is a repeat. Essentially, I think the one thing was it definitely validated the fact that culture-free direct from sample targeted sequencing is definitely possible, feasible. The very low indeterminate rates. I think the whole point or beauty of sequencing is if there is something to be, if a mutation exists, it will find it. There is sort of very little that at that stage that can go wrong. The call reproducibility also was shown to be very high. There's also the identification of mixtures down to 10% was shown by all three solutions. And in terms of the sensitivity, specificity, both genotypic and phenotypic, they did meet the TPP criteria for these drugs. Now I mentioned a set of drugs. Obviously, all of these solutions also looking to be able to, as targets get clearer for the newer drugs, these should also be covered by these solutions. And I think that's always going to be the goal. And hence the mutations catalog becomes an integral sort of methodology or an integral tool really for diagnostic developers as well to ensure they are being able to update their own assays, their own bioinformatics in keeping in line with the updated versions of the catalog. So really, and coming down to really this, the main part of the focus for this entire workshop, really, the advantage in TB compared to a lot of other pathogens is that the predicting of phenotypes from mutations is made simple because of it's so conserved, so highly conserved in terms of the genome. The expression is so reliable, very high specificity that you're seeing, and also mutations in sort of a set number of genes, about 15, 16 genes, really confirm most of the known drug resistance today. So I think there's a lot to be said in terms of I don't have to go out and search a lot of this, but I still need, it's conserved enough for me to be able to put this together. So what has been missing so far has been this asystematic sort of approach to put them all together in one place. So you can imagine to do that, there's a lot of studies, different people have contributed to various really good studies that even manufacturers have used to inform their assay development. But what is really needed is, can you imagine the largest, possibly the largest collection of data that's needed to inform this? And that has really been a big focus of the project, and really the team that will also be presenting on this call is setting up this whole global TB knowledge base, right? To be able to inform it with the largest data sets ever that exist for TB with phenotypic, genotypic, DSD information. So the cryptic being a big one of them, WHO surveillance, country surveillance data being another big part of it. And there's a lot of other contributors that if they are not already members of one of these, they are contributing through other ways. There's the legacy reseq, there's other projects through which coming, and this is something we'll continue to in a way seek out and continue to welcome. So if there's data that you are generating in your own studies, definitely welcome that. It needs to be, in fact, you want this to be part of this knowledge base so it can inform essentially the updated versions of these catalogs to ensure new mutations, emerging mutations, and even the confidence of each mutations is basically assessed over time and really optimized over time. And I won't even get to the methodologies because we have all the experts speaking about that. But what I wanted to emphasize was how this all fits together, because this is the process where we're getting as much of the data as possible globally to be able to get to this, the statistics that can then be done to identify each of these and to classify the mutations, if you will. And so, as I said, so all of this data is a lot of all of the work that happens in terms of a knowledge base, the analytic pipeline, the getting in the data in, being able to get the surveys in, being able to then eventually be able to also view and browse and see dashboards. But right now, this focuses piece here is the mutations catalog, as I said, which is the key tool that came out of it. Not even going to spend more than a few seconds here, but this is the very first global TB mutations catalog to predict drug resistance, as we said, far from genetic data for TB. And it's really being seen, as we've been speaking to more groups and even outside of TB, very much seen as a pathfinder, because you can imagine something, a very similar tool for other pathogens of AMR concern. Amount of data may not be the same, the amount of sort of the mechanisms of resistance may not be the same. So there's a lot more work to be done for those pathogens, but the very same, like sort of the similar methodology approach and the concept really is applicable beyond. And I think that's been one of the great things we've been able to do with TB, where there's clearly a huge need to be able to do this. So we're glad to be able to start this and hopefully can expand basically the utility or the methodology's utility further. And then a little bit on really, as we're talking, really coming back to sort of the DRTB and the diagnosis aspect. How we are sort of seeing this as an outlook for DRTB, really about in the future being able to use sequencing to guide for treatment decision support. And really here, as we see it, hopefully pending the NGS guidelines, hopefully that's coming in the future based on all the clinical work and the different data, not just this project, not just Seek and Treat, but other projects, other clinical evidence from other sequencing projects, to really be able to make end-to-end NGS solutions reality to detect genetic variants, and the variant interpretation or the mutations part, the RNS calling, really being driven by something like a mutations catalog, really setting that standardized or the reference standard there. And then really linked to the treatment algorithm, which really, again, the treatment guidelines that are updated to really make sure the diagnostic side is keeping up with the treatment side as well, to be able to get to a decision that's really takes into account all of this, these data points and inputs. So just as closing, where we are currently is that the clinical evaluation, as I said, is ongoing for the end of 2022 review, hopefully all going well, given the pandemic. The other part, we have almost finalized implementation guidance that I think Nazir will speak to a bit more about. Another aspect we're starting to work on is some of the operational guidance for countries, because a lot of countries are already deploying or have sequencing. And as I said, they're getting it through for other diseases as well, to be able to start to see how our countries to help be able to advise or guide them, or even be able to get input from countries that have done this really well, to learn from them. How can we share, and what are your best practices, or what are things that have worked well for you that other countries could learn from? And then the last one, really, there's been a capacity mapping work, which is really on leveraging COVID-19 capacity. As I said, there's so much sequencing capacity that's been built up for COVID-19. We've seen that with all of the variants, with the latest variant as well, because there's strong sequencing in some of these countries, which is where it was immediately the way recognized, detected. And I think being able to utilize sort of that capacity and that sequencing knowledge and even the technical knowledge that exists today to really capture it also for TB is going to be critical for us to make these technologies really work beyond. And so the mutations catalog, along with all of this work, is really something that will take this much further to make it a scalable and sustainable solution for countries. That I'd like to thank you and thank the wonderful team at FIND and all our partners. And thank you very much to Unity and WHO as well. Thanks.
Thank you very much, Anita, for a very thoughtful presentation, very interesting presentation. We will have time for questions after the next presentation. So I really invite everyone to type your questions on the chat. Next presenter for this afternoon, or this morning, or this evening, I don't know, is Dr. Nazir Ismail. Nazir is the team lead for TB diagnostics in the global TB program here at the WHO.
Thank you, Matteo. Thanks for the introduction. Let me just go full screen. Can you confirm that you can hear me and see the slides correctly?
Yes, perfect.
Okay, excellent. Thanks a lot, Matteo, and good morning, good afternoon, good evening to everyone. And thanks, Anita, for the great introduction. So it leads in well to my presentation on the use of next-generation sequencing for the surveillance of drug-resistant tuberculosis. I have no conflicts of interest to declare. So to start off with, I think just to set the scene, I think what's important to appreciate is the key strategy that outlines our response to TB, and that's the NTB strategy, which has set specific targets for us to achieve by 2035. And on that agenda is drug-resistant TB. And relevant to our discussion today is just specifically the drug susceptibility testing. And what's set out is drug susceptibility testing related to rifampicin, fluoroquinolones, and to some degree isoniazid. So the expectation is that every patient that's diagnosed with TB should at least be tested for rifampicin, and all rifampicin-resistant TB patients should be tested for fluoroquinolones. And then it's very well recognized that ionase resistance testing is important, but largely not detected. So the table at the bottom of your screen provides data from the Global TB report in 2020, so data from 2019. I haven't put the latest data, but I think the pre-COVID data is probably more easier to interpret, to be quite honest. So what you'll notice here is that this surveillance data provides us the estimates of what the burden of drug-resistant TB is out there. Of the 9.9 million global TB cases estimated, 465,000 were estimated to have rifampicin-resistant TB. But only just over 200,000 of those individuals were actually notified to be on a rifampicin-resistant TB regimen. And secondly, what you notice is the large burden of INH-resistant rifampicin-susceptible TB, which is estimated to be just over 1 million of the roughly 10 million TB cases globally. So what we clearly see is that there's a high burden of drug-resistant TB. What's also important to appreciate is that the surveillance data provides us an important benchmark of where we need to be, not where we are. And that's the importance of surveillance. But having said that, and looking at the NTB strategy, although rifampicin, INH, and fluoroquinolones are priority drugs for surveillance, what's very clear in terms of the regimens for treatment of drug-susceptible and drug-resistant TB is the emergence of new drugs and repurposed drugs that are moving to the front of therapy. And therefore, more comprehensive forms of drug surveillance are needed. And secondly, also for people that are treated with rifampicin-resistant TB, often more than 5 drugs are used in therapy, and therefore broader surveillance of drug resistance is important. So what is surveillance? And surveillance is defined as a means can be defined as the systematic ongoing collection, collation, and analysis of data for public health purposes. And secondly, the timely dissemination of public health information for assessment and public health responses as necessary. So there's 2 components to surveillance. The one is gathering the right data that can inform a response. And then the second is to basically ensure that that data is not just collated and collected and synthesized, but also used to direct public health responses. So it plays a very critical role in the response of any program. In terms of surveillance systems that are out there, it's either one of 3. The first is continuous surveillance. That's usually from routine testing, where there's a very high coverage of testing for a specific disease or resistance profile. The second is periodic surveys, and these are really aimed at sort of filling the gap where routine testing is not universal. And then the third is sentinel surveillance that's really very targeted to answer specific questions or to monitor and provide an early warning system for broader surveillance to be initiated. And what you see on the map on the right is around rifampicin resistance surveillance and the sources of data for the estimates that are provided. And what you'll notice is a large proportion of the sort of high burden countries, the countries, priority countries for TB, most of the data comes from surveys that really informs what's really critical to the response as such. So as I indicated, the surveys provide an important source of information. And what you'll notice from the countries that you've seen in the brown shading in the previous slide, a lot of the surveys have been conducted in the last 5 to 10 years. If we look at the colors of on the diagram, providing fairly recent estimates of drug-resistant TB. So the surveys have been pivotal in guiding our understanding of the magnitude of the problem, but also directing what we need to do. But the major challenge with surveys has been the reliance on phenotypic drug susceptibility testing, which proves quite challenging around the logistics, the quality assurance, and also what can realistically be achieved, the number of drugs that could be tested. Surveys have moved on towards using molecular WHO recommended rapid diagnostic tests, such as Expert, etc. And more recently, the use of next-gen sequencing, so sort of aligning with what is routinely being done for care in many countries. So what's the advantages of next-gen sequencing for surveillance? So as I indicated, often with drug-resistant surveys, sometimes you're fairly limited in what you can test based on phenotypic methods. And each drug you test costs an additional quantum of money. The second is that for many drugs that are of importance, typical DST may not be totally reliable. Additionally, and what you see in routine care as well is heteroresistance is emerging as an important problem. And next-gen sequencing provides that opportunity to assess the magnitude of that issue. The other important thing, as we move to more molecular diagnostic tests routinely being used, understanding these resistance conferring mutations and distinguishing them becomes quite important, and sequencing therefore sort of becomes an important backbone of routine care using molecular diagnosis. What has been a challenge is that often a lot of the surveys have been dependent on culture. And as Anita has highlighted, obviously with targeted next-gen sequencing, you can go to a culture-free solution as well, which is really enticing. And then the issues around culture itself, as I've highlighted, are obviously a problem. There are some additional benefits which are often not achieved through routine surveys in terms of using next-gen sequencing data for molecular epidemiology. And that can be quite insightful to also understand the burden of transmission, etc., which is critical for drug-resistant TB. While the startup cost for NGS can be quite steep, the actual cost relative to a phenotypic DST may not be that large in terms of direct costs. The big question is around the use of next-gen sequencing for surveillance, understanding that the prevalences could be quite low in many different settings. And this was sort of a pioneering paper by Matteo, who's chairing the session today, that looked at survey data from 7 countries and compiled that together. So it was basically drug-resistant surveys that were conducted, and in addition, a whole genome sequencing was performed. And what you can see in these figures, the red shaded and the blue shaded bars, are nearly completely overlap. And the figure that you see on the bottom left is for rifampicin resistance prevalence. The middle graph is about INH resistance prevalence in both rifampicin resistant and susceptible. And then the figures on the right are related to fluoroquinolones for Oflox and oxyfloxacin. But what's very clear is that there's clear overlapping bars, indicating that next-gen sequencing, at least based on this work, performed very well and could actually could be recommended for use for surveys as such, and has now become an important step forward in this field. So what is the existing guidance on the use of next-gen sequencing for TB? So the first guidance came out in 2018, sort of being the first effort at trying to provide information to countries and users on next-generation sequencing technologies. And that also included sort of the first sort of crude catalog of mutations. The second has been the latest guidelines that have been updated for surveys and surveillance for drug-resistant TB, and that has been updated and providing concrete guidance around populations, approaches to surveys and surveillance, etc., and becomes a cornerstone for surveillance activities around drug-resistant TB. And then as this workshop is really focused on the catalog of mutations, it's important to understand the context. And a lot of discussion will go on around the catalog of mutations today. So I won't spend too much time on that. But just to highlight that when we look at the catalog of mutations, it also provides an important surveillance tool in itself. It basically provides the mutations that are known to confer resistance to rifampicin first and second line drug resistance. So provides us a sort of a surveillance at a mutation level. Also provides an estimate of prevalence and the emergence of mutations, which is important, and as we've seen from, like, for example, the Swatini strain. With the data that we have in the catalog, we also have the clades that provide some insight on the clustering of drug-resistant TB. And as any surveillance is aimed at, it's not just for gathering data, but also guiding future use, and therefore these catalogs obviously provide a response strategy around development of new diagnostic tools. And in the longer term, it's really aimed at providing information that will inform patient care once the clinical trials have been done and we can evaluate the performance for clinical use. As Anita has highlighted, linked to all of this work is the database that underpins the catalog. And this really is aimed at being a global knowledge database to support the use of next-gen sequencing technologies and sort of managed through the monitoring and evaluation unit at WHO. It's a cloud-based collation of raw sequencing data and associated phenotypic and other metadata And as we'll hear later on around the standardized bioinformatics pipeline to develop this catalog really provides some robust methodology. However, in terms of surveillance, what's underway is to ensure that this database becomes useful and available publicly. So a dashboard with some analytics is being developed by Find and colleagues. The catalog has been released, but we're aiming to have this more visually presented and providing an exploratory tool for users. And then the most important thing is really around the longevity of the database and associated outputs from there. It's really the input and output of data, and a portal for automated uploading and downloading of data is being developed. One underlying issue that we're trying to address is ensuring that the data can be rapidly made available publicly, and we're trying to overcome disincentives for sharing. Obviously, we often get data fairly late, and trying to address those gaps in how we deal with the knowledge base. So what's coming up? So we've indicated all the different publications that have come out guiding and directing the use of NGS. A new guideline is aimed for release in 2022 on practical considerations for implementing NGS in surveillance programs, in national TB programs. This really will map out the intended use of NGS, providing a sort of direction of how to go about it, identifying issues that need to be addressed for country readiness, the whole cycle of supply chain, etc., to ensure that this could be implemented robustly, how to assess site level readiness, understanding quantities and the infrastructure required, validation of protocols to ensure there's standardization, and there's some quality assurance in the way it's delivered. And then obviously ensuring that there's some local capacity in being able to use and understand, interpret, and actualize next-gen sequencing. And then lastly, obviously, the framework to measure the impact of NGS DST. And this really is meant to be a lead up to its longer-term use in clinical care. And obviously, NGS for surveillance will still continue in the background. The target audience for this document would obviously be the NTP program, Ministry of Health, partners, donors, and obviously the ref labs and routine labs as well. So quickly, in terms of NGS implementation for surveillance, there's numerous benefits. I've already highlighted these, but it's really primarily around drug-resistant TB surveillance. But there's numerous other benefits once implemented around outbreak investigation, and molecular epidemiology beyond just the resistance profiles that will be identified. The guidance will provide sort of structured approach to how to go about implementing NGS for countries. So the country planning, the regulatory issues, the training around that, SOPs, data recording, reporting, quality assurance, to really get NGS into countries. And as we've heard around the SARS-CoV-2 work, this provides an ideal opportunity to leverage that gap. So quickly on the last additional component that's in these guidelines are some workflows, draft workflows to guide the introduction of surveillance in countries. So, we know from past experience from DRS surveys, some countries might not have fully developed DST capacity, phenotypic DST capacity, and some do. And then algorithms for those that require DST and those that don't, both upfront molecular tools and then either culture positive with phenotypic DST or not. And then the workflows that are split between targeted NGS and whole genome sequencing. So one is more culture-free, one is culture-dependent. But the important implication, obviously, the biosafety requirements when you require culture and phenotypic DST as part of the surveillance. Right, so in conclusion, I think what's very clear, surveillance plays an essential part of the public health response. It informs new strategies and provides a monitoring evaluation of changes that have taken place. Next-gen sequencing is clearly highly accurate for public health surveillance of drug-resistant TB with many practical advantages. The WHO guidance that has come from 2018, 2020, this year, 2021, has expanded over time to really guide countries in adopting technologies that are available. We will be releasing the new guidance in 2022 that will provide more concrete implementation steps for implementing NGS for surveillance. The catalog of mutations, which we focus on today, is an important source of surveillance, but also assist in future clinical use of NGS and guiding the development and understanding the use and contribution to this catalog and this database will be critical going forward. And then lastly, implementation of NGS really requires careful planning, and this latest guidance will really provide steps and pathways to implementation. So with that, I'd really like to thank the WHO colleagues that have been involved in a lot of the development work for this, but also our partners with FIND that have contributed in developing some of the material, the mutation catalog team, and many, many others that have contributed to this effort. So with that, thank you. Thanks a lot.
Thank you very much, Nazir. Very interesting presentation. There are lots of material that we didn't share, there are lots of information. Thanks also to those who have put some questions in the chat. I think there are a couple of questions for Anita and also a couple of questions for Nazir. We have 10 minutes for this discussion. So maybe I'll start rapidly with Nazir since I see you on the screen. So the three questions I see are basically quick one on differences between active and passive surveillance. Now, if you can just say a few words about that. Second question is about whether there is when the sequences that are available in the WHO database will become available for use by researchers, and whether there is any pipeline which is sort of endorsed by WHO for this kind of analysis. I think these are the three questions I'd like you to focus, Nazir. Thank you.
Okay, great. Excellent questions. Thank you very much. So maybe I'll deal with the second question, the last question first. So the sequencing and sequences and the availability. So of the sequences that were included in the report that was published, I think 92% of those sequences are already in the public domain. So they exist in the repositories, either NCBI, EBI, others, etc. So that data is available. We are working on, as I indicated, a portal to ensure that you could sort of identify it and have the phenotype, genotype all linked together and get that information collated, which is something we're working on. But in terms of the raw data, that is available. But going forward as well, we will aim to ensure that whatever data we receive could be made publicly available and encourage people to donate as far as possible. Obviously, one thing we will do, obviously, if we do not have permission to share it ourselves, we will definitely provide access to let you know who shared the data, and you can contact the primary donator of the data to us as such. So hopefully that clarifies the question around sequence data availability. The issue of pipeline endorsement. So that's a tricky one. We don't necessarily endorse a particular pipeline, but for the purposes of standardizing the catalog of mutations, a standard pipeline was required. And that included a huge engagement with a variety of stakeholders to try and input on the way that pipeline was derived. And we've made it sort of publicly available. So if you read the report that's been produced, I think half of the report describes the methodology, giving you all the parameters, etc., all the details of how that pipeline was set up and all the parameters that were included. So that does provide what we had done. I mean, there's a lot of manufacturers that have their own pipelines, etc. And we cannot necessarily predefine that. I mean, each person believes their pipeline is better than the next. So we're not here to choose one over the other, but we've collated people to provide a pipeline for standardized analysis, and that's what we have. So hopefully that clarifies. We don't necessarily endorse pipelines per se, but we've provided what's required. And the knowledge base runs on a specific pipeline. And if you do find, I mean, you believe that there might be some things that we've missed or things that could be improved, it's really meant as a global resource. It's not only ours. So if you've got any bit of information or advice or improvements, let us know. We'll share it with the team been working on the development, and we can always improve. So just to let you know, we're busy with version 2 catalog, and there's already been feedback on version 1. So we will obviously improve based on any feedback that we receive. And then the first question was related around active and passive surveillance. I think that's an excellent question. Ideally, active surveillance is always better in a sense that you have a targeted question, you systematically collect data, and there's less bias that's introducing what you get. So that's the ideal, right? And surveys are designed with that active process, choosing your population, choosing, powering it accordingly, etc. So that would be the ideal, but often comes at quite a lot of money and cost. I think passive surveillance on its own does provide important data because it sort of naturally comes, right? You just have to do the sequencing on isolates. And if you look at the catalog, was not purposely designed. We collected whatever data we could receive, right? We didn't go and design country studies for them necessarily, right? So I think we're using passive surveillance, but it provides important data. It identifies frequencies, it identifies important mutations that are known or unknown, their associations, etc. So we'd encourage anyone to undertake passive surveillance where it's easy and doable. But ideally, we'd like structured surveillance going forward in terms of having a short questionnaire, providing additional data. So the data we collect becomes far more meaningful and can be far greater in terms of its impact. I hope I've answered the three. Those were the three, right? Okay.
And thank you very much, Nazir. I think it's extremely precise and really good answers to the questions. There are, as you see, more questions, so maybe, Nazir, you can answer them in the chat or later. I suggest I move on to a few questions for Anita. One of them is actually a question from Andrea. Khabib, it's about the project that you are managing at FIND and whether the outcome could be an approval of a specific TNGS solution or an approach, basically. That is one question. And the other one, I think, if you have a few minutes to comment on, is from Enos Mazzini, which actually is something that you mentioned in your presentation. It's about whether we can capitalize some of the investments done on COVID-19 on WGS or NGS for TB. So the two questions. Thank you, Anita.
Yeah, really good question. So yeah, first one from Andrea. So we are planning to submit all the three technologies, hopefully the performance of them. So it's not a comparison versus each other, but really comparison against the reference. So as many of them that basically can meet the criteria will go in. And of course, the answer on what WHO will be looking at, I will let Nazir answer. I'm hoping it is also about the technology. It's three different tools, three different, in a way, brands or products that are really towards a technology. Nazir, maybe you can just mention that one.
Come again, sorry.
For the WHO review process, would be approval of a specific TNGS or more on the methodology or the method?
Okay, so in terms of WHO recommendations for clinical use of TNGS, we would provide a class-based recommendation as we've moved on for current technologies. And they would ideally aim to provide a very similar design and performance to make sure that they meet the class requirements, but we won't necessarily recommend one particular technology. However, there's not many, to be honest, in the pipeline currently. I think you all have done a lot of work on that. But whoever performed within the expected performance bracket and provides obviously the qualitative and cost data, then we would make a recommendation accordingly, but it'll be class-based.
And the second one on COVID, yeah, that could be, I'll try not to talk for hours on that, but you're absolutely right. There's so much investment on COVID related in sequencing and everything else, infrastructure that should really be leveraged. So one thing we're doing, at least to start on this process, is to do the surveys with countries. So at least firstly our project countries, and then really going beyond to understand where the sequencers for COVID are set up, like literally where they're placed, because a lot of it is also seeing programs and where the access to samples will come from. So that would be your first basis with at least starting with a core group of countries and then expanding to see where the sequences are placed, what else can be run on the sequencers, how easy it is to be able to really programmatically utilize them, because technology-wise, that is ideally and suitably set up exactly for that purpose. But programmatically, whether that's achievable today or some changes need to be made to ensure that can happen is still something we at least need to see, and hoping with WHO we can map out some of this, and with many of you on this call as well. So we'll be reaching out on some of these questions to help you optimize the investments you have in sequencing in your countries. Thank you.
Thank you so much, Anita. I see that there are more questions in the chat. I encourage everyone to keep asking questions, that we will have time later on for a longer Q&A. And also some of the speakers can already provide some answers in writing in the chat. I suggest we move on, because we have a very heavy agenda, and move on to the next session and the next speaker, who is Dr. Tim Walker, who works at Oxford University, who will be talking about genotype and phenotype association studies for drug resistance determination. I'm sure Tim is already online. Thanks.
Thank you, Matteo. Hello, everybody. Yes, I've just got 15 minutes to talk through our thoughts regarding association studies between phenotypes and genotypes. There's a short quiz at the end. I'm afraid, given that we're on Zoom, there are no prizes to be handed out, but we'll go through the answers at the end. I've got no conflict of interests other than a receipt of grants from charitable and governmental organizations. So why do we do genotypic phenotypic association studies? Well, as we've heard already, the phenotypic DSD hasn't really done the job for the patients around the world who need it most. And there is great hope that genotyping is going to solve the problem. It's faster, it's probably cost saving. And as we'll see in a moment, it's arguably more consistent as well. But if we use phenotypes, we need to be able to, genotypes, we need to be able to interpret them, and therefore we need to do some association studies with phenotypes. And to be very clear, what we're doing with a catalogue is trying to predict the phenotype through the genotype. People often ask why we don't predict clinical outcome. It's because it's an extremely difficult thing to predict, because there are very many confounders, and it would be extremely expensive to do so, even though it would be an optimal thing to do if it were possible. So we use phenotypic DST as a proxy. So I'm going to talk about some phenotypic considerations, some genotypic considerations, what to associate, some confounders and traps that we need to be aware of, how to think about significance, and then how to manage expectations. And I won't linger on any of these because it's a quick talk. So just to start off with phenotypic considerations, broadly speaking, you've got two ways of looking at phenotypes. You've got a binary phenotype, which gives you either a resistant or sensitive outcome, and you've got a continuous phenotype in the form of a minimum inhibitory concentration, or MIC. We chose to analyze binary phenotypes, but this, although on the face of it, this might seem to have been a simpler approach than analyzing the MICs. The panel on the right-hand side will show you that, in fact, there was great complexity even in this, because just for one drug, you can see this panel is relevant to just isoniazid. in the very many samples that we analyzed, we had to cope with numerous different methods, culture methods, and numerous different critical concentrations that had been deployed. And to manage the many different combinations, we had to try and generate a hierarchy for these data whereby we would weight results that were generated by WHO endorsed methods and critical concentrations over those that weren't. And in some cases, we actually had to exclude some phenotypes because there was insufficient information around them. So even if one tries to do a simple analysis around binary phenotypes, one's still faced with great complexity because of many different approaches that are out there. If you look at MICs, the picture becomes even more complicated, largely because you end up dealing with unendorsed methods. And also, when you translate a catalogue that's based around MICs into clinical practice, you really need to revert back to some form of binary phenotype, because in the end, what the clinician wants to know is, can I give the drug or can I not? But even when you're dealing with phenotypic binary phenotypes, again, things remain quite complicated in some instances. So these are some nice cartoons generated by Claudio, who will speak after me, that show the wild type distribution, the MIC wild type distribution here in green, and where there may be a resistance mutation, you can see that the distribution of MICs in this particular instance does not overlap with the wild type distribution, and in such circumstances makes it quite easy to draw a cutoff between the two, and to one side you have sensitive, to one side you have resistance. But in quite a lot of instances, life isn't quite so simple, and you end up with resistance mechanisms that have an MIC distribution that actually overlaps with the wild type distribution, as you can see in panels B and C. So when you're then dealing with a binary phenotype and an isolate has a particular mutation where the MIC distribution for that mutation overlaps with the wild type distribution, you can end up in a situation where every time you test the sample, you get a different result because it can fall either side of the cutoff. So even dealing with binary data can be problematic. The genotypic considerations. So again, broadly speaking, two initial considerations. Do we look at the whole genome, or do we look at a set of candidate genes? So the whole genome has about 4,000 genes. A cartoon of that is depicted on the right-hand side from this publication down here. And one approach to analyzing that would be a genome-wide association study. We instead opted for a targeted gene approach. So we took, for each drug, we looked through the literature and chose the genes that were supported by pre-existing data to be involved in drug resistance. And as you can see from the table on the left-hand side, we divided those genes into two tiers, where tier 1 was genes that we had high confidence in that they are likely to be associated with resistance, and tier 2, slightly less confidence based on fewer data. The main advantage of this approach, of the candidate gene-based approach, is that you're enriching for your results, because your pretest probability or that any mutation is associated with resistance is far greater when you start with candidate genes like this. It also allows you to avoid the problem that you run into with GWAS sometimes, that when you do get a significant hit, you're not entirely sure which drug that particular mutation may be relevant to, because of the non-independence of phenotypes in TB. So, for example, you would often see isoniazid resistance in the setting of rifampicin resistance. So those two are epidemiologically non-independent, and that does make interpretation a little bit tricky sometimes if you're not doing a targeted gene-based approach. Other considerations would be whether to analyze the data at, excuse me, excuse me, at a nucleotide level or at an amino acid level. So for example, the most common mutation to confer resistance to isoniazid is the catchy S315T, that if we analyze it at an amino acid level, we would not differentiate between the different nucleotide mutations that can lead to that particular amino acid substitution. And there are two examples of those below. The other consideration is whether we consider the possibility that synonymous mutations in encoding sequences can cause resistance, and there's some evidence for this, for example, in a very nice publication by Yadon and others in Nature Communications in 2017, when they showed that a number of synonymous mutations in PNCA were causative of resistance to pyrazinamide. We, however, made the assumption that synonymous mutations don't cause resistance for the purpose of our analysis, for better or worse. And in terms of the nucleotide versus amino acid level, we chose to analyze at the amino acid level in coding sequences. Of course, this itself can be quite complicated because there are genes whose promoters actually lie in other genes. So if you look at INHA here, INHA has a promoter mutation that lies within FABG1. And now one needs to do a little bit of pre-analysis before one does the association study here to ensure that the particular promoter mutation in FABG1 is actually annotated as a nucleotide mutation and not as an amino acid substitution, or in this case, for L203L, synonymous mutation. But within that synonymous mutation, or parent synonymous mutation, is in fact only one particular nucleotide mutation that that does the business in terms of conferring resistance. So some interpretation of the genetics is important before one starts to make sure that all of the mutations are annotated appropriately. Next consideration is how one deals with mixed alleles, or the so-called hets, short for heterozygotes. So in a sequence, these are two cartoons I've just created, but you could see, for example, that the number of mutations in the left-hand panel that are mutant versus wild type, you can see a predominance of mutants, which is the Cs versus the Gs. On the right-hand panel, you can see that the Cs are a minority population. And you can imagine that the effect on the phenotype is going to change as the mutant changes from minority to a majority allele. So how does one handle these? And does one introduce a cutoff? Does one ignore them? It's complicated and difficult, and there's no easy answer to this. We ended up excluding het calls in the sense that we wouldn't try to generate associations for them, but we did take their presence into account when it came to analyzing other mutations, and I'll go through that in a little bit more detail in a second. Other considerations are about how to handle deletions or insertions. So broadly speaking, these can be frame shift or in-frame mutations. So again, just some very basic genetics, but a frame shift mutation would be an example of that might be the insertion of a single nucleotide that then changes the nature of every amino acid downstream of it because you've shifted the reading frame, whereas an in-frame insertion would be the insertion of 3 nucleotides, so a whole codon, not affecting any of the codons downstream of that. So that you can imagine the structure of the protein and an in-frame mutation still has a chance of being relatively intact. So does one differentiate between these? Does one pool all mutations within the same, all insertions or deletions within a single gene to gain power, and making the assumption that they all disrupt the protein? Or does one analyze them all independently, which is what we ended up doing, even though there's an argument that insertions or deletions starting at the same locus in the gene are unlikely to behave differently. So again, here is an example, for example, in PNCA at position 518 in that gene, if you have a frame shift mutation of 1 or of 5 nucleotides, one would imagine that it would behave the same. For better or for worse, we ended up analyzing all of these independently. Now, having given some thought to all of those considerations, the next question is, what actually are we associating here? Are we seeking to just make associations between single mutations and the phenotype, or are we, do we want to look at interactions? So there are a number of different interactions that one can look at. Additivity would be one such interaction where 2 mutations occur together, and the effect is the sum of the effects of each of those. Synergy is another phenomenon where the effect is greater than the sum of the parts. And then thirdly, epistasis is an interesting phenomenon where the effect of one mutation is actually cancelled out by the presence of another mutation. So an example of that is the EIS promoter mutation that confers resistance to kanamycin, and does so unless there's a loss of function mutation in the coding region of EIS, in which case the promoter mutation doesn't confer resistance to kanamycin. So that's an example of epistasis. Now, our analysis was simple, and we just looked at single mutations. So we didn't seek to identify any of these phenomena, although arguably maybe we should have, and we will endeavor to do so next time. So having gone through each of those considerations, I'm going to quickly walk you through the initial algorithm that led to our analysis. So as I said, we took a candidate gene-based approach. We looked at both coding sequences and defined promoter regions for each drug. We made an a priori assumption that synonymous mutations were not associated with resistance and excluded those from further analysis. And we also ran some preliminary algorithms to classify other mutations as not associated with resistance before we started, and masked those. We then assessed each remaining mutation, and if across a whole set of candidate genes and their promoter regions, we were left with only one mutation. We defined that as a solo because it was on its own. And if the phenotype for at least one of those was resistant, we classified it as algorithmically on the basis of this algorithm as resistant. And then we repeated that process for all the isolates, for all the phenotypes, and amassed a large number of mutation, or for every mutation, we amassed a large number of phenotypes so that we would see that they were either found in R or in S isolates, and then we could perform some statistics on those results. We did the same for sensitive mutations. So these were ones that were only ever seen in sensitive isolates, or only ever seen in sensitive isolates when they were solos, i.e. the only mutation in town. And then we were left with a third category that we were unable to assess, and we called these U for unable to assess, I guess. And these were mutations that were never seen by themselves. So they were never solo, and therefore couldn't be isolated. Their effect couldn't be isolated. And where at least one of the phenotypes was resistant, because if a mutation never occurs by itself, but is always in a sensitive isolate, it gets classified as sensitive. But these ones were resistant on at least one occasion and never occurred by themselves, and they ended up as U. And they didn't make it further down to the route of statistical analysis. The next question we needed to ask ourselves is, what is it we want to know? And quite important, I think, is distinguishing between these two questions. So first of all, how confident are we that a mutation predicts resistance? Important question. And how confident are we that it predicts resistance with a given frequency? So there are some mutations, as I showed, based on Claudio's nice MIC distribution figures, that will confer resistance consistently, inconsistently, if you like. So if you have if the mutation confers an MIC that's very close to the critical concentration, then every time you test a sample with that mutation, you might expect a different binary phenotype. such inconsistency can be consistent in the sense that you know that you're going to get that inconsistency if you re-phenotype the sample, and you might end up with a result that says this particular mutation is 50% associated with resistance, but if you have very tight confidence intervals around that 50%, you have a sense of how that mutation is going to behave in the future. So it's important to know how confident we are that it predicts resistance with a given frequency. It's just as important as it is to know the answer to the more simple question, which is how confident are we that a mutation might predict resistance. Now, there are some confounders and traps in all these analyses. So first of all, population structure, and those of you who are familiar with GWAS will know that a good GWAS will control for population structure, and what we have done doesn't. So this means, for example, there might be a lineage effect. So lineages here are colored, but it may be a lineage or a sublineage that is intrinsically resistant, or indeed that is only found in a country where treatment guidelines have ended up selecting for resistance to a particular drug. And so those effects aren't seen in our candidate gene approach, and we may end up as a consequence associating the wrong mutations with the phenotype. That is a danger. Having said that, the vast majority of drug resistance mutations have evolved recently and therefore occur at the tips of these trees rather than in deep-lying branches where they would affect population structure. We need to be aware that there are compensatory mutations. So one example of this, for example, is AHPC promoter mutations that compensate for CAT-G mutations. And I think there was at least one example where we missed a large deletion within CAT-G, and the only mutation that we saw was an AHPC promoter mutation. Now that may lead one to assume that it's the promoter mutation, the AHPC compensatory mutation, that confers resistance. No, it's not. The follow-up question to that is, does it really matter as long as it consistently predicts resistance? Given that we're in the game of predicting resistance here, one might argue that that's not such a great danger. Sample mislabeling is something one has to be aware of. There is an intrinsic error rate to everything we do, including phenotyping, and there's a background rate across most studies of about 2% mislabeling. So that has to be borne in mind when interpreting any of the results. And we've spoken about MICs. So very briefly, statistical testing. How does one present the results? There are a number of different ways one can present them. You can look at odds ratios, PPV. We ended up presenting all our data as using Fisher's exact tests. And then how does one control for or deal with the false discovery rate? Of course, the number of, if you do many tests, you're going to accidentally encounter some that are significant, or falsely significant, if you like, and one needs therefore to correct for multiple testing, and one has a choice of using a rather conservative approach such as Bonferroni, or maybe a more pragmatic approach, which is what we chose using the Benjamin-Hochberg approach. Very briefly, this simulation just shows that the prevalence of resistance in your whole data set is also relevant to the interpretation of the statistical outcomes. So for example, where you have a very low prevalence of resistance, so 0.05% phenotypic resistance in the whole data set, and a mutation that's seen 100 times will have a high odds ratio, as colored indicated by a color here, even though the proportion of mutations of isolates that have that mutation that are resistant might be as low as 20%. Where the prevalence of resistance in the data increases to 20%, then that same mutation would require penetrance, if you like, of up to 80% before it gets into the red, where you get a high confidence, a high odds ratio. So knowing your data set allows you to better interpret the odds ratios. Now the same is of course true for Fisher's test and any other tests. Just if you take 10,000 samples and you have a different prevalence of resistance among them, 0.5 here and 20% on the right, and a mutation that's seen 100 times, and 10% of those mutations are seen in resistant isolates, you end up on the left-hand side with the mutation being enriched for resistance with a low p-value, and on the right-hand side actually being enriched for sensitivity with a slightly also low p-value. So one needs to understand the data one's working with and interpret these findings in the context of all of that data to really make sense of the stats. And finally, expectations. We'll never be able to predict with greater sensitivity than the phenotypic error rate. So if you've got a basic baseline intrinsic phenotypic error rate of 2%, you're never going to get a sensitivity of greater than 98%. And that's worth understanding, otherwise we end up chasing rainbows. The other consideration here is that there are different drugs behaving in different ways, and we know more about some drugs than we do about others. So there are maybe marginal gains to be got from doing ever more analyses on first-line drugs, whereas there may be great steps still to be taken when analyzing new and repurposed drugs. So that's all I have to say. There's a short quiz now, which I think a panel will appear. Here we go. So I'm going to give you 3-4 minutes just to quietly go through these 6 questions in your own time. So you can scroll up and down and select an answer, its single best answer for all 6 questions. Once those 2-3 minutes are up, or once people have answered, most people have answered, we can talk through the answers to those questions. So I'll be quiet for a few minutes. So I'm aware that I talked for a little longer than I should have, and I see that people are still answering, but I wonder whether we can start going through the answers. Feel free to answer the questions we haven't got to yet in this little discussion in the meantime. So the first question was, what was the genotype compared to? And the majority have got it right here. So the answer is B, a binary phenotype, R or S. So we chose not to associate to MICs in this analysis, although that may be done in version 2. So answer is B, the binary phenotype. Question 2, what type of mutation did we not assess? So again, the majority have got it right here. The answer is C. We made an a priori assumption that synonymous mutations in coding sequences do not confer resistance. We did assess all of the other options. Question 3, what is epistasis? So again, the majority have got it. I've got the answer right here. So the answer is A, one mutation that cancels out the effect of another. And I gave the example of the EIS promoter mutation being cancelled out by a loss of function mutation within the EIS coding sequence itself, relevant to kanamycin resistance. Question 4, what is a solo mutation? So the solo is the jargon that we've used for mutations that where we find a mutation to be the only mutation, only non-synonymous mutation or promoter mutation across all concatenated candidate genes. So the answer therefore is D, which a slim majority got right. Question 5, what does it mean when a mutation is classified as U? So this is a mutation that is never seen by itself as a solo, as just described, and where at least one is associated with a resistant phenotype, so that it doesn't end up getting classified as S. So the answer here again is D. And again, the majority got that right. And finally, our findings could be confounded by, and the true answer is C, population structure. So again, the majority got that one right. So the idea here is that the population structure isn't being controlled for and therefore can act as a confounder. Although I suspect because of homoplasy, in other words, the fact that most mutations arise multiple times and have arisen recently, and therefore at the branch tips, the population structure actually doesn't end up causing us too much trouble. I'm happy to answer any questions, any follow-up questions, or to explain any of that in more detail in the chat, but we should probably move on given the time. Thank you for thank you to everyone for having a go at those questions.
Okay, hello everybody. I hope you can all see my screen. So the purpose of my talk is really to bridge what Tim has just introduced you to and to set you up for Paolo's talk, who will be walking you through the products, so to speak, of this entire analysis, namely the catalogue in the form of the Excel file. I have a couple of conflicts of interest to mention, the most important being that I work as a consultant for BD and that I used to work as a consultant for QuantityX, which is a diagnostics company. So just a quick reminder, we covered all of the drugs for which there currently are critical concentrations. So this meant that we couldn't cover cycloserine because there are no currently endorsed breakpoints. For protonamide, there's also no critical concentration, but please be aware that there's a paper currently under review that will present a lot of the data that will be needed to set a breakpoint. So these data should be coming soon. And the other drug that we couldn't cover were the carbapenems because we have no idea how to test them and what the breakpoint would be. And we only covered kanamycin and caprimycin really for historical interest. I mean, neither drug is endorsed for the treatment of patients any longer, but kanamycin in particular was helpful because it provided some insights for amikacin because some of the resistance mechanisms are shared. So the final product really was to stratify mutations into 5 different groups, which are here, group 1 to 5. And group 1 and 5 are really groups where the expert committee deemed that there was high quality evidence that these mutations were associated with resistance, in other words, could be interpreted as a marker for resistance to the drug in question. This is shown here in red, or on the other hand, group 5 that were not associated with resistance. So in other words, if you found one of these mutations in a drug resistance gene, then you could ignore that as being irrelevant in terms of resistance. And probably the best example that you might be familiar with is CAT-G codon 360 something is a very frequent, deeply rooted mutation that does not confer resistance, or in CAT-G at codon, in GRA at codon 95. In addition to that, we had category 2 or 4, where the evidence for a mutation being associated with resistance or not associated resistance was there was less evidence compared to group 1 of 5, but where the committee nonetheless felt that they should be listed as interim markers of resistance, and therefore to gather more evidence and to improve primarily the sensitivity for drugs such as pyrazinamide, where we have lots and lots of mutations, and therefore if we had only used group 1 mutation, the sensitivity would have been fairly poor. And this leaves us with group 3 mutations, which are mutations of uncertain significance. And these are mutations where basically the evidence was inconclusive. So we couldn't classify them. They didn't meet the criteria for group 1, 2, 4, or 5. And more data will be needed to classify those going forward. So it's important for you to appreciate that essentially we took a 2-step process to classify mutations. We had an initial confidence grading, which is essentially based on the association between a mutation and phenotypic DST data based on the data that Tim Walker just introduced to you. And so this gave us an initial grading, and this was then subsequently modified using a series of criteria that I'll introduce to you in a moment, to get a final confidence grading, which is essentially what the final outcome of this whole exercise is. So just to show where our samples was from, I mean, we took effort to try to sample or get isolates from that are as globally diverse as possible. And that's really important for 2 reasons. One is, even though Mycobacterium tuberculosis complex is very homogeneous compared to other bacterial pathogens such as E. coli or a Klebsiella, there is still relevant, even pre-existing biological diversity, even before you introduce selection pressure by antibiotics. So therefore, it's important that we sample strains from different parts of the world. And of course, the second reason for sampling strains from different parts of the world is that we want the associations to be as representative as possible, because you can have TB population is very clonal, so you can have some strains that are dominant in one part of the world, but very rare or not found at all in a different part of the world. And again, in version 2 of the catalogue that was mentioned before, we'll try to make this analysis even more representative than it currently is. Table 1 of this is taken straight out of the WHO report, is that provides an overview of the number of isolates that were either the total number of isolates or isolates that were resistant, either that had a phenotype that met either current or previous WHO criteria, and that's denoted here by WHO label, or isolates that had any phenotype shown here by OR. And as expected, we had the vast majority, we had the largest number of data points for isoniazid, simply because that's an old drug and traditionally one that was frequently tested. And also because it is a first-line drug, the rates of resistance was high, so around 35% or 12,000 isolates that were resistant. Whereas the number of isolates for which we have data was drastically reduced for the neuron repurpose drug. The lowest number was actually for delaminate. And what you can also see here, that the composition of data were quite different. So we had very few data points for WHO-endorsed methods. For delaminate, for example, the vast majority of data points actually came from non-WHO endorsed methods. And also, so not only did we have fewer data points for these drugs, but also the rates of resistance as defined using various methods was a lot lower. So for instance, for bedaquiline, the rate of resistance was just 1%. And that's a function primarily of the fact that this is a recently introduced drug. And as it's more widely used, you expect the rate of resistance to increase over time. So, as I mentioned earlier, the primary classification was based simply on the phenotypic data and the genomes based on the sort of scheme that Tim introduced just now. But of course, but then we needed to apply some other criteria to sort of modify the initial classification. So the first criteria which Tim already hinted at was that we had to weight based on the quality of the phenotype. So for example, there might have been the highest quality phenotype would be a method that's currently WHO endorsed with a currently endorsed critical concentration, which would have trumped a phenotype with a critical concentration that's no longer WHO endorsed. And then that would trump a mutation that was classified based on a method that was not WHO endorsed. And crucially, when we go through the mutation catalog with Paolo, you'll find that it's completely transparent as to whether what the initial classification was and whether it was a mutation was then up or downgraded, and based on, and it's also clear based on which criterion or criteria this occurred. The other criterion we had to consider was sort of an algorithmic criterion as to whether it was assigned by pass 1 or pass 2, and I won't go into detail about that. You can read up on that in the catalog. Or mutations were weighted based on the type of resistance gene that we looked at, because as Tim mentioned, there were some genes for which there was a lot of pre-existing evidence, often also with allelic exchange or knockout experiments, that for which we had high confidence that mutations were causative, whereas for others, the a priori evidence was weaker. So we would upgrade and downgrade mutations accordingly. The other important point to appreciate is that obviously this mutation catalog did not exist in isolation. WHO has obviously endorsed multiple previous catalog, rapid genotypic AST methods, such as the various line probe assays or the Cepheid assays. And so and it also published a sort of preliminary list of mutations in the sequencing manual. And we wanted to really capture all of these various decisions to make this catalog a one-stop shop. So for example, this diagram here shows the rifampicin resistance determining region in RPOB. And you can see here the different probes that cover this region by WHO-endorsed assays. And there are specific mutations that are covered by mutation probes by line probe assays. And if these are found, these would be reported as resistance detected by the assay in question. And we wanted to make sure that any mutation that is interpreted in this way by one of the rapid tests that you, most of you would have used for many years, would then also feature like this in the catalog, unless there was evidence to contradict that. In other words, we wanted to make sure that irrespective of which assay you use, that you end up with the same interpretation, which wasn't always the case previously because some of the previous decisions hadn't been particularly well integrated. Other important previous WHO decisions that we considered here was one related to the so-called borderline RPOB mutation. There was a lot of controversy and also confusion in the field around 7 specific mutations that confer only modest MIC increases and therefore have poor reproducibility when you test them. Now, this in the past was also exacerbated by the fact that the critical concentration for RIF and 7H10 and MGID was too high. This was recently corrected. But even with the new critical concentrations, you can retest the same strain multiple times in your laboratory and sometimes test resistant and susceptible because the breakpoint divides the MIC distribution, as Tim mentioned at the beginning of his presentation. And WHO in the recent systematic review of breakpoints for rifamycins had introduced specific rules that these 7 mutations should be interpreted as a marker of rifampicin resistance, irrespective of the phenotypic result. And therefore, we wanted to make sure that this was reflected in the current catalog. WHO had also introduced an expert rule whereby any mutation except synonymous ones in the rifampicin resistance determining region should be interpreted as a marker for rifampicin resistance. And then we also assumed complete cross-resistance at the critical concentration between levofloxacin and moxifloxacin. The other expert rule that was endorsed was really regarding mutations that are required for the activation of a few drugs or their inactivation, and therefore any insertion or deletion of premature stop codon was assumed to lead to a loss of function phenotype and consequently drug resistance. And therefore, this expert rule was applied for EDHA, for ethionamide, JIT, for streptomycin, cartionazanide, and PNCA, and pyrazinamide. And this is really to help us to improve the sensitivity, because often you could have an isolate that had only one or two isolates with one particular indel, and we'd never have sufficient statistical power to really classify these mutations. These would have ended up as being in group 3 had we not used this expert rule. And last but not least, we relied on the literature to some extent. So the report wasn't a systematic review of the evidence in the literature, but we did rely on the literature for key mutations that we know would have caused problems otherwise. So for example, there's a mutation at codon 90 that's covered by the WHO-endorsed second-line LPA, but also the XDR Cephid cartridge that is very frequent in the Uganda genotype in Central Africa. And this causes a systematic false resistance result. And we would not have been able to classify this mutation as not causative of resistance using our analysis, but we did want to include it so that countries that do encounter it would know how to interpret it. But again, we tried to avoid this as much as possible to really derive all of these mutations de novo and avoid bias, given that a lot of studies often featured very few isolates. So I'd just like to walk you through a couple of drugs, and it won't go through all of the drugs because you can explore those in detail, but I just wanted to sort of give you an example of what the output at the end is. So I wanted to start with rifampicin because most of you have an intuitive understanding that rifampicin resistance should be relatively easy because RPB is an essential gene, so therefore the number of resistance mutations is relatively limited. And that's true to some extent, but actually you'll find that the picture isn't quite as easy as you would take away by reading some of the literature. So just as a reminder, this analysis was based on 34,000 susceptible isolates and 10,000 phenotypically resistant isolates. And with the exception of 2 mutations outside of the rifampicin resistance determining region, all of the approximately 130 mutations that were associated with the resistance definitively on an interim basis fell into the rifampicin resistance determining region. And you can see that 24 mutations that met criteria for group 1 gave us a sensitivity of 92% and a really good specificity and positive predictive value. But that 106 out of the 111 mutations that were classified as marks of resistance on an interim basis were classified based on the expert rule that any mutation except synonymous mutation should be a valid marker of resistance. So you can see that, first of all, even in a gene that is essential, there is a large number of different mutations, and a lot of these are very rare indeed. But including all of these mutations only sort of bought us an additional 4%, which is obviously important for rifampicin, but also shows you the importance of the expert rule. And you might say, oh, hang on, this expert rule seems to be very adventurous. But actually, all of you, those of you have been using either GeneXpert or the first line LPA have been de facto using the expert rules for years, because obviously these assays cannot differentiate the vast majority of mutations in those regions. They can only tell you whether there's a mutation or not. And so you then effectively assumed that the mutation that was detected was a valid marker of resistance. So you've been using the expert rule all along without explicitly labeling it as such. And of course, there may be exceptions to the expert rule. In other words, a non-selling mutation that generally does not confer resistance in RRDR, and there might be 1 or 2 examples of that. This will really be the focus of version 2 of the catalogue, to avoid overcalling resistance based on the expert rule by simply excluding individual mutations. Now, I think just a quick word about pyrazinamide. So I think pyrazinamide is sort of the typical drug that most of you would think about a complicated drug, because it's a non-essential gene, so therefore you have a very large spectrum of mutations that could confer resistance in principle. And indeed, we had over 100 mutations where we had high confidence that these are marks of resistance, but these only gave us a sensitivity of 57%. And so we had to use some expert rules regarding loss of function, but also sort of relaxed criteria to classify mutations, which then allowed us to classify an additional 230 mutations, but that really only increased the sensitivity by 15%. So this tells you that there are lots and lots of mutations that are very rare, but these still have an excellent specificity. So, and because WHO appreciated that, even though this represented an important improvement over previous guidance in the sequencing guide, WHO endorsed an additional expert rule whereby any group 3 mutations or any novel non-synonymous mutation should be interpreted as a marker for resistance if it occurs in an isolate that is resistance to rifampicin, because in those circumstances, the pretest probability that it confers resistance is very high. And this expert rule is really endorsed to help you with dealing with the novel mutations if you find it in a rifampicin-resistant isolates, which is the subgroup of isolates for which sequencing would most likely be used. Now, the big sort of limitation of the catalog was really that, for instance, for beta-arginine and clofazimine, we were able to classify mutations as not associated with resistance. In other words, mutations that can ignore in terms of the interpretation, but we weren't able to classify any mutations as markers of resistance. Now, this doesn't mean that mutations in ADPE or RB078 are not markers of resistance, but it does mean that in our particular analysis, we didn't have enough statistical power to classify them. Whereas for linezolid, we were able to classify one mutation, which had, and that's in RPLC, and this is globally the dominant resistance mutation, which in our data set has a sensitivity of about 40%, but looking across study probably accounts of for a sensitivity of about 50%. Sort of, I just wanted to flag one point in that EIS-14 and RS-1402, which previously was only recognized as a marker for kanamycin resistance, was also now recognized as a marker for amikacin resistance. And the newly endorsed Cepheid XDR cartridge already interprets these 2 mutations correctly. And the European Laboratory Initiative is due to update its interpretation guide, which you can find on this website here, that also has an accompanying online course. And we're due to update this interpretation guide so that you can implement this change in your day-to-day practice if you indeed rely on the second line LBA. So as Tim already hinted at, I think there's certainly room for improvement. So a key focus for version 2 of the catalog will be new and repurposed drugs. And because the primary difficulty with there was that many of these mutations are rare. And in addition, many of these confer only modest MIC increases. And therefore, if they flip-flop between being susceptible and resistant, it's more difficult to classify them. And so we'll be focusing on these to really provide a better guidance on these. And in addition, we might have to think about endorsing either LX criteria for classify these rare mutations, or also endorse sort of expert rules regarding loss of function mutations for genes that have a large spectrum of resistance mutations, such as RB078, which is definitely the resistance gene for beta-arginine, but that we couldn't classify as part of this analysis. We'll have to focus on exceptions to expert rules because we want to avoid overcalling resistance in certain settings. We also need to improve the approach that we use for compensatory mutations, because mutations that are compensatory mutations, in other words, that are secondary to the primary resistance mutation, are still valid markers for resistance, but they don't ordinarily show up as solos, and therefore we weren't able to classify them. So even though AHPC upstream mutations are interrogated as a marker for isoniazid resistance by the XDR cartridge, which is certainly correct, we weren't able to classify them as part of this catalog. And that's something that we need to work on. We need to stratify the level of moxifloxacin and isoniazid resistance because we simply performed as at critical concentration, particularly for moxifloxacin, going to stratify into high and low level resistance. And I think more importantly, we need to provide sort of better integration with how this catalog should be used. In other words, should phenotypic confirmatory testing be done? And if phenotypic testing is carried out, how do we deal with discordant results? In other words, how do we overrule the discordant results for which there's already clear guidelines for RIF, but for which equivalent rules are needed for the other drugs, particularly for the fluoroquinolones, but also bedaquiline. This particularly really comes down to mutations like the yellow one here, where because of the technical variability in phenotypic testing, you can have the same isolate flip-flopping between susceptible and resistant results simply because of technical variability. And in those circumstances, actually finding the mutation is a more reliable marker for resistance. And so we need to provide clear guidance. So thank you very much for your attention. We will now move on to the poll. So I'll give you about 3 to 4 minutes and then I'll start running through them. Okay, in the interest of time, I'll start going through these questions. So question answer B is the correct answer for question 1. So for the purposes of the interpretation, we chose a fairly conservative analysis approach for this version 1 of the catalogue. But this meant that we weren't able to classify RV078 mutations, even though some but not all mutations in these genes do confer resistance. So there's a difference between whether a gene is involved in resistance at all versus whether we had enough evidence using the approach in this particular analysis to classify them as markers for resistance. So for question 2, there's a fairly even split across all of them, which is interesting. This is also probably the most difficult question. So the correct answer, so the statement that does not apply is C, because essentially there's a difference between whether something can insertions and deletions and premature stop codons, because for those, even if they occur in an isolate that is otherwise susceptible to rivancin, there's a high pretest probability that it confers resistance. So those would be interpreted as a marker for resistance, irrespective of the resistance to other drugs. Whereas non-synonymous mutations, you'd only interpret as a marker of resistance if they occur in an isolate that is also resistant to isoniazid. And again, there's a whole section in the report that explains in detail the reasoning behind this particular rule and with references. So question 3 is really related to the expert rule to loss of function mutations. So WHO did endorse an expert rule for PNCA, ETHA, and JIT for loss of function mutations, but not for RB0678 so far, even though in the vast majority of cases, frame shifts in these genes do confer resistance. And again, this will be the focus of version 2 of the catalog. The only other point that we'll also have to consider in this context is that epistasis is possible in this particular case. In other words, if you have an inactive RB678 gene, it can only confer beta-glucophasine cross resistance if the efflux pump associated with it is active, which is not always the case. But again, WHO will provide guidance in that context. So the next question was regarding some of the other statements, and the statement that did not apply was B, which most of you got right, because essentially any mutations, with the exception crucially of synonymous mutation in RRDR, should be assumed to confer resistance to rivancin. And indeed, there can be synonymous mutation in this region, which can be frequent in some settings. So it's important that synonymous mutations are excluded. And currently, at least for this gene, there's no evidence that these could confer resistance. And this leads me to the last question. And here, the D is the correct answer, because here the main challenge is really that mutations that confer large MIC increases are easier to classify, because these should test reliably resistance at the critical concentration, and therefore you need fewer data points to confidently associate them with resistance. All right, thank you very much for your patience. As I said, there's a lot of detail that we provided in the report itself that you can read. The slides will be shared at the end of the presentation. It has my email address. You can reach out to me or some of the other speakers if you have any questions. And again, don't be shy about questions. There's no such thing as a stupid question, it's only stupid answers. We'll take care of those. And it's really important that we also improve the way we present the results based on your feedback, because we want these results to be used. So if you find any ambiguities or so, or have any suggestions, they'll be gladly taken on board. Thank you very much.
Thank you so much, Claudio, for that fantastic presentation, and Paul, and thanks very much, everybody, for engaging in those questions and answers. Very much appreciated. Okay, folks, and thank you also, Tim Walker, for that, for your presentation and the questions. Okay, folks, we're going to take a 5-minute break. We're going a little bit long here, but that's totally fine. So we're going to take a quick 5-minute break just to give everybody a chance to get up, take a bio break, and then we'll be back. So we'll put the recording on pause while we're taking a break. And if we could see everybody back here promptly in 5 minutes. It's really important, folks, that you don't log out just because logging back in requires you to be brought back into the room. So if you could just put cameras on and mics on mute and come back in 5 minutes. So it's 6 minutes past the hour here where I am. So I'd say let's come back then at 11 minutes past the hour. Make it exactly 5 minutes. So 11 minutes past the hour, we'll restart up. Thanks everybody, much everybody, and we'll be starting with Paolo Mioto's talk directly after the break. Okay, everybody, we're going to get going again here. Hope everybody is back. We're just going to jump straight in, keep us on time here. Thank you, everybody who's still with us after this. These wonderful talks. So the next speaker we have is Dr. Paolo Mioto. Dr. Paolo Mioto is a senior scientist at OSR in Milan, and Paolo is going to be talking a little bit about assessing and using the catalogue of mutations, the practical stuff. Paolo, away to you.
Thank you, Tim, for the kind introduction, and I will start sharing a few slides. Okay. Okay. I hope everybody can see my slides. So good morning and good evening, good afternoon. And during this presentation, I'll provide an overview on how to access and use the catalog. I have no conflict of interest to disclose. And during these first 10 minutes, I will cover how to read the catalog tables, where and how to find information within the catalog, and I'll drive you through a few exercises using the Excel file. First of all, I would like to remind that the purpose of the catalog is to provide a standard reference interpretation of mutations and provides the identification of neutral and resistance mutations, plus further details on the frequency of the mutations. There are a few display differences between the Excel tables and the table you find in the PDF catalog. Basically, the PDF tables are reporting main columns, whereas in the Excel table, you have all the details. And in particular, here you can see we have the drug column, the variant or mutation column, the columns reporting the observation in RNS isolates, and the derived calculated sensitivity and specificity. Then we have the main criteria used for the initial grading that, as previously described, are the PPV solo and related 95% confidence intervals, lower and upper bounds, the PPV, the odd ratio solo and associated statistics. And please note that in the PDF tables, we reported only graded mutations. Those we omitted the number of solo observation that is anyway available in the Excel file. Finally, in the PDF tables, you find the dataset driving the grading and additional grading criteria such as expert rules and the final confidence grading that is the most important output of the table, of course. In the Excel file, you can see additional details. Of course, there are the 95% confidence intervals for all the statistics reported, plus the likelihood ratios. And also you can find additional details about WHO previous guidance about the mutations here, and whether a given mutation is covered or detected by currently endorsed molecular DST. For a quick consultation of the catalog, you can look for your mutations in the PDF tables, and here is a brief overview of what you have. As mentioned, you have the drug of interest, the mutation, and the numbers for false positive, true false negative, true positive, and true negative, leading to the sensitivity and specificity calculated and reported in the tables. And plus all the values that are used for the grading of the mutations. And finally, of course, you have the final grading criteria. Throughout the catalog, we used a color code where values reported with a specific color meant that they met the threshold for the grading system. So for example, any mutation found as a solo in susceptible and resistant isolates equal to or above 5 is reported in red, and you can find the same scheme in the flowchart reported in the PDF catalog explaining the grading criteria. Similarly, this is, for example, for PNCA, where we applied, let's say, relaxed thresholding for pyrazinamide resistance. You can see here that the PPV column can be reported in yellow when the PNCA mutation for pyrazinamide show a PPV above or equal to 50%, and this is a step of the flow chart of the grading system. This also applies for interim association for neutral mutations, again in a PNCA for pyrazinamide, and in general for neutral mutations that are reported in green in this case. So there is a clear relationship between the flow chart and what you have in the table in terms of colors. In the PDF tables, we can find all non-synonymous mutations classified as associated with resistance, not associated with resistance, or interim, and in also premature stop codons graded by the statistics and thresholds. Indels and premature stop codons classified as associated with our interim by expert rules are available in the Excel file. And as we will see later, the Excel comes with two spreadsheets called one mutation catalog and the second genome indices. Variants classified as uncertain significance, genome indices, and synonymous mutations are found in the Excel file only. So now I will ask you to open your Excel file that you downloaded at the link shared in the chat. And I would move on with the Excel file. So here is how you see the file. And you can see here you have 4 spreadsheets, basically. There is the mutation catalog and the genome indices that I previously mentioned. And for each of them, you have also a spreadsheet reporting a legend for all the abbreviation you find in the catalog. So as you can see here, the spreadsheet is pretty extensive in terms of the number of columns. And you have all the information I mentioned before. You have the number of observation in R and S isolates, the PPV solo values that are used for the grading scheme, plus the 95% confidence intervals, the sensitivity, likelihood ratio. Here we have the odd ratio, and the statistics in this case is reported on a dedicated column. And you can see here that again, you find the same color scheme I mentioned. Then we have the initial grading confidence, the data set leading to this initial grading confidence. And finally, all the information, the additional information that brings us to the final confidence grading. I will start you showing a few examples. Let's start with something really simple. You can either decide to filter for the drug first or look directly for your mutation in the variant column. And I will look for the RPOB S450L mutation. And here you go. Here we have all the numbers related to the mutation. It has been found as a solo in more than 5,000 isolates. And since this is meeting the threshold on this category, it is highlighted in red, as previously mentioned. You have all the numbers that brings you to these sensitivity and specificity and likelihood ratio. And as you can see, we met the criteria for the PPV solo lower bound that is reported in red, and so on for the odd ratio solo and the statistics. So according to these thresholds, the mutation was associated with resistance as initial confidence grading, and the evidence was brought by both the data sets based on WHO endorsed methods only, and the old data set that includes also WHO non-endorsed MIC data. As you can see here, I can enlarge a little bit. We have additional information. This mutation was previously reported as associated with resistance by the WHO guide published in 2018. And also it has been reported as associated with resistance because in the rifampicin resistance determining region, in the rifampicin critical concentration guide published earlier this year. And you see here, you can find that there are several WHO endorsed tests that target this mutation. And the final confidence grading is reported here. I would like to show you another example on RPOB. That is the I491F mutation. This mutation is a borderline RPOB mutation, and it is highlighted in purple in the mutation column. And in this case, again, it's associated with resistance. You can see here that this is a borderline mutation, and all borderline mutation for RPOB by WHO definition have been highlighted in purple, so you can easily find them. And you have the final association. If you are in trouble, please note that me and Claudio will be available for another 20-30 minutes after the seminar. So don't panic. We will be available for detailed answer questions. I would like to show you a couple of examples of mutations that are not associated with resistance. Sorry, I forgot one point. So I would start with the CatG R463L. That is a marker of genetic background. And in this case, for example, you see that this mutation is not meeting the PPV solo criterion, and so it is not red in this case. And you can see here that the mutation is flagged as neutral masked on this column in green, and the mutation is finally graded as not associated with resistance. And similarly, if you look for the GRA S95T mutation, we have two entries because this gene has been targeted for both MOXIE and MOXIE and LIVO. And again, this is not expected to be associated with resistance. Indeed, there are no flagged columns except for the one of the odd ratio, but the odd ratio alone is not sufficient for associating a mutation with resistance. And again, you can see here that the mutation is flagged in green as not associated with resistance. And also in this case, the mutation is considered not associated with resistance by some of the currently WHO endorsed methods. I would like to show you one example of an interim association. So for example, we can take the PNCA H51P mutation. And here you can see that for PNCA, we have yellow entries for on the present solo in resistant isolates only, and the PPV value instead of the PPV solo. And this is a mutation that is associated with resistance interim by evidence. So there is no an expert rule that is upgrading the association, but this is just driven by the data. And we applied these relaxed thresholds to the PPV and the number of isolates with the mutation. Another example of interim association can be given by the RPOB S450Y. As you can see, this mutation would not be graded by the algorithm itself, because the mutation has been observed only twice and the threshold is 5 instead. And indeed, the initial confidence grading was uncertain significance. However, since this mutation was previously reported by WHO guidance as associated with resistance because part of the rifampicin resistance determining region, we applied an expert rule here, and you can see the additional grading criteria, and the mutation is finally graded as associated with the resistance interim. So I hope I made clear that you can have interim association either by the evidence we have available for a mutation or by expert rules. Another example can be given by the ETHA W109 premature stop codon for ethionamide. In this case, again, the mutation is not meeting all the criteria because it is passing only the threshold on the PPV solo, but the number of solo observation is too low. Again, the threshold is 5. Here we have 3. And the initial confidence grading was uncertain significance, but given the expert rule on INDER and premature stop codon for loss of function mutations in the ATHA gene, the mutation was finally reported as associated with our interim. There is another criterion to define interim association, and we can look at the RPOB S450Q mutation, again for rifampicin. This mutation met all the criteria to be associated with resistance. You can find here it was present as a solo in at least 6 isolates. The PPV solo lower bound is above the threshold. The odd ratio solo and the statistics associated are are fine. So the initial confidence grading was associated with resistance. However, the association is coming only from the old data set that is including phenotypic DST, not endorsed by the WHO yet. And for this reason, the association in this case was somehow downgraded, and the final grading was associated with our interim. So again, depending upon the type of evidence, the final grading can change. Similarly, I would like to mention another example on PNCA, looking at the mutation I6L. This is for pyrazinamide. And this mutation, as mentioned, for pyrazinamide PNCA, we applied relaxed thresholds to define interim association with resistance or for not association with resistance. And in this case, you can see here that there are two blue entries for the PPV solo and the PPV solo upper bound in this case, instead of the lower bound as used for the association with resistance. In this case, this mutation is meeting the criteria for interim association with no resistance. So basically, the association is, the grading is not associated with our interim, since there is nothing else for this mutation. The final confidence grading is not associated with our interim. So as we have interim association for resistance, we have interim associations for no resistance. Now I will show you, of course, as mentioned, we have in the Excel file all the uncertain significant mutations that are not reported in the PDF file. And here, in this case, I filtered for uncertain significance by final confidence grading. And we have two main groups for these mutations. Mutations that were found in as a solo in SNR isolates in more than 5 cases that are highlighted in red, but are not passing one or more of the other thresholds we put on the PPV and the odds ratio. Or we have all the remaining mutations that were not found as a solo in S or R isolates, or were found in small numbers, let's say above, below 5. And indeed, they are reported all in red. So this means that the mutation is too rare, or it's too rare alone as a solo. Another thing that is reported in the catalog are in the Excel file are the combo mutation, and sometimes I receive email asking what is combo meaning. And basically, in this case, we reported some of the solo mutation happening in combination because it could be useful for designing diagnostic assays. And I will take as an example the typical CATG315T mutation plus the NHA promoter region. And in this case, it's here. So you have, as I mentioned, this combo entry in the final confidence grading. This is not a confidence grading at all. It's just mentioned. It is just to highlight that you can find the frequency of some combination of solo mutation in the data set. And as you can see here, the CATG mutation plus the C-15T INHA mutation was found in more than 1,000 resistant isolates. And this is the sensitivity of the combination of the two mutations and the specificity, etc. So it's just for reporting a few details on the combination of relevant mutations. And as I mentioned, this is not available for all the combination, but we focus this on some solo combination. What to be mentioned again? Okay, here is a column called genome position, and you will find the genome position of the mutation in this specific spreadsheet called mutation catalog only for promoter mutations, such as in the case of EIS, or nucleotidic changes in the ribosomal gene. This is not reported for the aminoacidic mutation because, of course, the genome position is related to the nucleotides and not to the amino acid. And if you are interested in looking at the nucleotides related to a given aminoacidic change, you have to switch to the genome indices. And you here have again the same variant column. And in this case, you have the genome index with the reference nucleotide and the alternative nucleotide column. So you can, let me just filter for any aminoacidic substitution. Let's make it random. So here you have this aminoacidic change in AHPC. Of course, the codon number is 12 because it's also reported here, and you have the exact position and nucleotidic change in the genome. So please remember that if you are interested in the nucleotidic changes, you have to switch the spreadsheet. And related to this, on these genome indices, this is a kind of super expert information. Of course, here you have all the nucleotides, and you have the columns also for the number of isolates found resistant and susceptible for a given drug and the related confidence grading. So you have not to switch between the two spreadsheets if you are interested in looking at nucleotidic changes. And after, of course, we have 3 columns for all the drugs. And at quite the very end of the spreadsheet, as I said, this is a kind of super user information starting from this column called final annotation reference. There are a few columns where we reported a tentative human genome variation society nomenclature for the variants, and this is supposed to be machine readable. So if you are a bioinformatician, you can use this to import the catalog in your systems. Of course, as I said, this is tentative and we are working on this, but we try to make this available for bioinformaticians as well. Okay, so I would move on with a few exercises. I will show you a question and I will do the exercise with you. Let's do like this. So then I will switch to the Excel. So if I want to see the grading of the variant C-14T in EAS, and look if it is associated with amikacin, kanamycin, or both the resistance, it's pretty easy. We can, I hope you can do it yourself. You can just type your variant here. And you see that it is for AMI and Kana. And at the very end, the mutation was associated with resistance for both the drugs, as previously mentioned also by Claudio. for let's do another exercise on ATHA. Please try to search for ATHA S399 premature stop codon and try to see if it is associated with our interim, because it is not present in S isolates, or it is present in R isolates only. We applied an expert rule, or we applied expert rule by literature evidence. In the meantime, I will go as well for the same mutation. I hope you are done. So this is the entry. You see here, initial confidence grading was uncertain. The final grading is associated with interim. So the answer to the question is because we applied an expert rule on premature stop codon and indels for ATHA. If you want to look for frequency of mutation, you have two options. The first one is, let's take that specific mutation, RPOP S45. Okay, so you can use, if you want to, of course, know in this the frequency in resistant isolates, you can just simply use the sensitivity. If you want to see the overall frequency, you can sum up the number of observations for the given mutation and then divide by the total number of isolates. So here you have all the numbers you need for do the calculation on the frequency of a given mutation. And of course, you can also calculate the frequency in susceptible isolates. I would like to show a brief exercise also on bedaquiline and clofazimine. So there are no variants associated with the resistance for these two drugs. Let's understand why. It is because none of the mutation caused cross-resistance to the drugs. All the mutations were found to be neutral. None of the mutations was found in R isolates, and none of the mutations was found in S isolates, or none of the mutations found met the threshold set in the classification algorithm. So basically, if we look to bedaquiline and clofazimine, you can see, let's start filtering for the first criteria, the criterion that is the presence of the solo mutation. So I can use the filtering color. Below threshold are all those reported in black. As you can see, then I can go to the PPV solo lower bound, that is the second thresholdings, and there is nothing in red in this column. So basically, none of the mutation found for these drugs is passing all the thresholds needed to be achieved, let's say, for being associated with resistance. So the correct answer is E. Let's do an exercise for nucleotidic changes. Please try to find out what are the nucleotidic changes for PNCA S104R mutation. So as I said, if you are interested in nucleotidic changes, you have to switch to the genome index spreadsheet. Search for your variant S104R in PNCA, and here is the answer. Basically, in this case, we have 3 different nucleotides changes that are leading to this mutation, and that's why for the amino acidic changes we cannot provide the genome position. Sorry, I have filtered for bedaquiline and other stuff here. Another thing that you need to remember is to reset your filters, otherwise you end up without finding what you are looking for. So you cannot find here the genome position, but the genome indices spreadsheet is showing the genome index and the nucleotidic changes here. Okay. Now there are, I will stop, no, I will keep sharing my screen because then I can answer the polls as well. I would ask Caroline to start the polls. There are 3 questions and I will leave you 3-4 minutes to answer the questions. So I'll start with the first. The mutation is RRS G14840. Okay. The mutation is, the correct answer is B, because it is associated with amikacin interim, because evidence is derived by the old data set only. We can go to the poll number 2. So as I mentioned, if you are interested in genomic index, you have to switch to the genome index spreadsheet. Let's search for the mutation. And here we have 2 nucleotidic changes. So the correct answer is D, because the genome indexes are reported here. And I see that most of you answered correctly. We can move to the 3rd question. I'll search for the first for the drug in this case, then I look for the mutation. So the mutation is unclear significance. And the correct answer is B, because this mutation was not meeting any of the criteria for the association. However, it was found in one BDQR isolate as a solo, and it was and if we look for other mutation in the same codon, you can see here that there is another isolate with a I2S mutation that is found in resistant isolate. Okay, with this I finished, and I'm just to recall you that after the session I will be available together with Claudia with a specific question for any mutation. Thank you. Over to you.
Thank you so much, Paolo. Great walkthrough of everything. And I see there were some folks that had a couple of little technical issues of things not showing up that they expected. As Paolo said, we'll have a session directly after this. We can directly work with you to try and make sure we get that. We're running a little bit late, so I'm going to keep the question and answer session very, very short to keep us finishing on time. I have one specific question that I wanted to give. Tim Walker answered it in the chat, but I wanted to give him a chance to answer it in person. The question was from Pakistan. Since mutations to bedaquiline for new and repurposed drugs are graded undetermined, how should one clinically interpret and communicate non-synonymous mutations in genes associated with bedaquiline resistance? I want to give you a chance, Tim Walker, to answer
that. Geez, thanks, Tim Rodwell. I mean, it's a great question, and it highlights the major problem in the work that we presented, which is that we just don't have anything to say about new and repurposed drugs. It does leave clinicians in a bit of a pickle. They got a sequence to hand, they got a patient in front of them, they need to make a decision whether to give the drug or not. The bottom line is that the WHO catalogue cannot guide you on that. So to some extent, you're on your own, unfortunately. Were I the physician in front of the patient and having to make the decision, what would I do? I guess I would first of all search the catalogue to see if the given mutation has ever been seen before. Have a look to see how many times it was seen in resistant and how many times it was seen in sensitive isolates. The chances are that those numbers will be small. I would have a quick look through the literature to see if there's anything else to be learned about that mutation from other sources. I would probably have a look at the other drug susceptibilities and see if I can make any inference about the probability of bedaquiline resistance given the resistance to other drugs on the basis of the sort of patterns of drug resistance that we see. And then I would probably give the patient enough drugs, a larger number of drugs, so that there's a decent probability that enough of them work. It's messy, but I'm afraid there's not a better answer than that at the moment.
Thank you, Tim. And with that, I'm going to ask a question, since there's no other questions from the group that I haven't seen, that I've seen. I'm going to ask all of you something that I get asked a lot, and there is just ahead of time, I'm going to ask 3 of our speakers to answer this question from their lens. There is no right answer, and I think that's exactly why I want 3 different people from different lenses to answer this question. So I'm going to ask the question of Nazir as a representative of WHO, Tim Walker as an acting infectious disease physician, and Claudio as an academic advisor to diagnostic groups. And the question is this: what should I do? How should I clinically interpret a resistant interim result? And I'm going to start with Nazir, with you.
Thanks, Tim. So I think in terms of the question is around resistant interim results for any of the mutations in the catalog. So I think it's not a straightforward answer, right? So you get multiple different drugs, the clinical context in which that isolate is identified. And the second thing, just to be clear as well, so I know you've asked it from a clinical perspective, right? Maybe let's just step one back before we jump into that question, right? So as it stands right now, currently, we don't necessarily recommend sequencing as a clinical tool for treatment, right? Our WHO's position is that it can be used for surveillance purposes. The issue is, I mean, it's not so straightforward. We need to look at the clinical evidence around the performance of the technology. So what we're looking at in the catalog was a very specific pipeline that was developed and used as sort of as a reference standard. But in clinical use of technology, we just need to be wary that different pipelines are being used by different people. So what we see is, first question is whether that detection is correct or not. And that's what clinical assessment, either the validations will need to do, right? The question of when you ended up with the interim resistance associated result, right? You need to look at what's the underlying reason for that. Is it due to use of a technology that wasn't WHO endorsed? For example, the cryptic plates that have been used as part of the catalog, right? So there, I think we've acknowledged in the report that, for example, for bedaquiline and for other drugs, right? Often for the newer drugs that we haven't had much data from the standard methods like MIGID and AGA, right? So in that sense, I think when you look at that result, then you'd need to interpret it based on the data that you see, right? Understanding that this is not a sort of a definitive association, there's certain levels of uncertainty, right? And would require, you'd still regard it as resistant, right? So maybe let's start off with that point, right? So you still regard it as resistant because we're not saying interim unknown, right? We're saying interim resistant. So it is considered resistance, but the certainty of that resistance is less than we would have for definitive associated with resistance, right? That's the first point. The second point is that if you're interpreting it as resistant, right, which you should, then it would obviously need additional clinical follow-up to make sure that the decision that you take needs to be contextualized. So obviously, depending on the risk factors of the patient, for example, I mean, you find an interim R-associated mutation, right, and the patient is rifampicin susceptible, right? It changes the whole dynamics. So I think, when it comes to the interim category, it is, yes, interpreted as R, but know that there's a level of uncertainty and it needs to be contextualized correctly. So I think maybe that's sort of the best approach for now. The last thing I'd say before I hand over to others is that bear in mind that the interim is not aimed at, interim means it's in between something at this stage. So obviously, as the evidence grows, the body of evidence, it is possible some of the interim would become associated with R, right? Definitively, it might have been just a statistical issue that we put it as interim, or it might have been like maybe some of the expert rules that Claudio spoke about. So it's a mixed bag when you're looking at the interim, right? And I'd say that it needs to be contextualized as such.
Perfect. Thank you. A very loaded question, which is why I'm asking for 3 very different lenses so that you can all answer from your lens. And that was a perfect answer. Thank you. Claudio, you're next. As an academic who's often advising diagnostic companies, how do you personally interpret? And this, again, I'm asking for your personal view, not representing an institution in your particular case. What's your personal view of what RI means to you?
My view is that, it's not my view. The committee report them as interim resistant. In other words, I would interpret it as do not use that drug in the but with a few caveats. There could be some cases, obviously, for fluoroquinolones where the level of resistance matters. There could be some WHO regimens for which resistance to a drug is not an exclusion criterion. So it has to be taken in the context of what treatment decisions you can actually make, I would say. But on the whole, I would say, well, you base your initial treatment decision based on R and avoid the drug unless resistance to the drug is not an exclusion criterion for the regimen, or you can adjust those, for example, to overcome, say, low-level resistance, which is would be the WHO position. But,
so if I was a commercial company and I came to you and I said, I'm making my interpretation, I'm rewriting my interpretation catalog after the WHO mutation catalog came out. I'm rewriting my interpretation insert for my, the Hain, say if you had name your company comes to you and says, I can either say it's R, or I can say it's S, or I should say it's RI. What should I do as a commercial company? What would be your recommendation as a commercial company that they should put in there? I would
use the language that, so I wouldn't report an RI as R. Okay. So I would have that nuance in it, but obviously it is, I mean, that's, I think, one of the, personally, I think there needs to be better guidance as to how to use the catalog, because at the moment it stands there and obviously everybody sort of fills in what he or she wants to a certain degree as to how it's used. And I think there needs to be greater clarity, because again, if the initial decision then also matches a phenotype downstream, then everybody is happy. But then also, as I mentioned earlier, the problem really arises if you get discordant results. And so I think this needs to be integrated more effectively with downstream treatment decisions. But I mean, we said RI for a reason, because we felt that there was enough evidence to base an initial decision based on that result. That would be my take on it.
Okay, got it. Tim Walker, you're an ID doc. You're an ID doc on the floor, and somebody says to you, "Hey, I got this mutation." Says RI. What should I do?
So I don't disagree with anything that Nazir and Claudia just said. What would I do? First of all, use a different drug. RI is R for clinical purposes. Having said that, if your options are limited and there isn't a good alternative, next step then is to say, well, can I get round this? Can I still use this drug? And to answer that question, I'd ask two additional questions. One is the one that Nazir first mentioned, which is, why is it interim? Is the underlying evidence supporting this as R strong or weak? And secondly, if it does look convincingly to be an R mutation, can I overcome it with a higher dose of the drug? For some drugs, one can do that.
Got it.
Again, going slightly off piste there, but as one so often has to in these situations.
Exactly what I wanted. Some of the nuance from these three different perspectives, very much appreciated. I see there's two more questions that have jumped up. Nazir, can we go on a couple? Can I quickly answer those, or should we go? We have time to answer them. Okay. First question is from Eric. How do we address the lack or an inadequate data for Group A drug resistance? What are the chances of existing resistance to this drug in the community? That's going to be a difficult one to answer. I am going to hand that one to. I'm going to give that one to Claudia.
So just remind everybody. So Group A drugs would be levo, moxi, so the fluoroquinolones, bedaquiline, and linezolid. So this will be, bedaquiline and linezolid will be the focus of version 2 of the catalog. So fortunately, linezolid resistance is exceptionally rare globally. So that's in a way is a good thing. And then half of that we can already account for. I think bedaquiline is the drug that gives me the greatest headaches out of the entire list for two reasons. One is we have a large diversity of resistance mutations, so similar to PNCA in a way, but we have epistasis, so sometimes a mutation, the exact same mutation can confer resistance, sometimes not. But I think the biggest problem for bedaquiline is that, with exception of ATPE mutations, most mutations confer only modest MIC increases. So even if you set your breakpoint correctly, phenotypic testing isn't a reliable confirmatory test for your mutation. So all of these factors come together, which make bedaquiline problematic. And on top of that, there are some settings where you already have fairly high rates of pre-existing resistance. And we've seen that from multiple settings now that resistance is selected for fairly rapidly during treatment. So I think for version 2 of the catalog, we'll have to think very carefully about how to craft, the right kind of thresholds for these drugs, how to use expert rules to try to improve the sensitivity for the catalog. But it's I think that to me is the biggest challenge, and one that where we'll have to be, I think, creative, because if we don't do anything, I think we'll just find that bedaquiline resistance increases and goes under the radar. So we'll need to come up with pragmatic solutions for this particular drug. But yeah, this is, I think, by far the most difficult. Whereas for linezolid, I think with a focused push, I think I would expect that we can get the true sensitivity up to 80% or more. I think the biggest problem for linezolid is that given that it's so rare, the positive predictive value phenotypic testing is actually really poor. So a lot of what we think is of linezolid resistance is actually just a false resistant result of simply because the proportion of random errors is fairly large compared to the true prevalence of resistance.
Great, thank you very much, Claudio. Yes, another thing that, just so that you know, one of the things that we're trying to do with version 2.0 is we're trying to take sort of focus in on specific collections that are made specifically to highlight these resistance to these particular drugs, so that we can try and improve our association algorithms. That's one of the things we're trying to do next time around. Okay, one very last question. This I'm going to put to you, Nazir. It sounds like a surveillance question. How to validate low prevalence RIF resistance in a particular area, for example, in an environment where a neighbor might have very high prevalence resistance?
Yeah, so I think the issue of prevalence needs to be defined correctly. So the thing is, what you're saying is there's two different areas that are neighboring with two different prevalences. The challenge is if you're using routine data and you end up with, for example, in one area, you're only testing high-risk people with previous treatment that end up getting rifampicin resistance. You find a high prevalence, but in another area, you're testing all TB patients. You would expect to find different apparent prevalences, right? So depending on the risk group that you're investigating. So the first thing is to confirm, the methodology that's used to define prevalence, right? And the population that's being included, because that creates some degree of variability. So if you look at the way surveys are done, usually it's a very systematic collection of, individual participants in a study that basically would define their previous risk history, and then make sure that the methodologies being used for resistance detection are the same. So there's a lot of standardization to really define prevalence. So for example, in one area you're doing, for rifampicin, you're testing with AGA, the other one you're doing midget, and there's a high prevalence of the 511 mutations. You would find again differences. So I think, validation of prevalence requires a sort of epidemiological study to define that resistance. And usually for surveys, it's normally population-based, not sort of a convenient sample at a hospital, etc.
Thanks very much, Nazir. Very thorough answer. So with that, everybody, I'm going to call the primary meeting to a close. And I just want to thank, first of all, World Health Organization for hosting and making this work available to the public. I want to thank our speakers, Anita, Nazir, Tim Walker, Claudio, Paolo, as well as my co-chair, Matteo, and a huge thank you to the WHO team for running this, Caroline and your whole team there for all the organization that was needed to make this work. So thank you very much. Also, of course, to the participants. Thank you all for taking the time out of your, what I'm sure are extremely busy days to come and listen to this presentation and more importantly, be a part of this presentation. So with that, the official workshop is closed, but we still have a session after this with Paolo and Claudio for anybody who wants to sort of talk through examples, ask specific questions, say, hey, why couldn't I get this button to do what I thought it should do in the Excel, etc. We have that session coming up. And with that, I'm going to turn up to Nazira. Oh, sorry.
Yeah, I think we also like to thank Unit 8 and find the response to the session as well. And then there's a query around the certificates. So maybe I don't know if the union people can just answer that question before we break out. So
yes, thank you.
Answer that question.
Thank you, Nazia. So once the Zoom meeting will close this, so we encourage everybody to answer the question to the survey, which will appear in your browser. And you will receive follow-up email in the next few days, so with a link to view the recording of today's event. And as far as the certificate, they will also be sent by email within the next week. And they will be given to everybody who has attended 2/3 of the workshop and who is answering the question from the evaluation survey. Thank you very much.
Thanks a lot. So we'll leave you in the hands of Paulo and Claudio. Well, whoever's had any problem and had any difficulties, please stay on and we can help you out with the catalog. We know it went through quite fast, so if there's any, we'll just spend a bit more time with those that had some trouble as well. I wanted clarity.
Great. Paolo, probably the first one is to maybe deal with that issue, see if we can help with that issue of some folks who weren't able to find a specific mutation.
Yes, I think that if you look for the mutation in the mutation catalog spreadsheet, since it is a synonymous mutation, you cannot find it. So there are no matches in this file, so you have to switch to the genome index file. And so if you look here for the silent mutation, you are able to find it. It was I can't recall the number now, probably 8L. It was one of the questions, so let me go back. Oh, sorry, it was the GFP L103L, and here it is. So be careful for synonymous mutation to switch to the right spreadsheet. Are there any specific questions about the catalog? Please drop your questions in the chat.
So I'm not seeing any specific questions.
One has just come through. It's about how often the catalog will be updated.
Oh, okay.
So we are working on version 2 of the catalog. And as we mentioned earlier, the main focus will be really the new and repurposed drugs, for which the sensitivity in this present catalog was low. But especially for things like nitromidazoles, where we have 6 non-essential large genes spanning multiple kilobases, this is going to be a never-ending story. So we'll keep on finding novel mutations. And again, also for nitromidazoles, there appear to be 2 to 3% of strains that have baseline resistance just by chance, not because of exposure. So that will be a never-ending story, which will require updates every now and again. I don't know how long-term that will be funded, but it certainly will be needed. Maybe Tim or Nazir, you could comment on that.
Yeah, so I think the plan is to update this as we've termed it regularly, right? But obviously it needs to be needs-based. So it's very clear that, as Claudia indicated, there are certain gaps that we were aware of, and like with the new and repurposed drugs. So the intention has been to update it now. In the future, it'll all depend on need. Our longer-term vision, which we're working on currently, is to ensure that the catalog would become live with a curated sort of check over time. What I mean by that is, for example, a lot of the work that's been conducted thus far, taking all the genomic data, running the bioinformatics on it, and then doing the association studies have required a lot of manual curation. And the plan currently is to make sure that all of that can be automated to a large extent. And that could become sort of a real-time dashboard where people could see as they donate data into the system what's changing. However, what we've learned as well is that although that live system would be great, there's a lot of nuances that experts like Claudio, Paolo, Tim Walker, etc., really need to sort of provide some oversight and definitive analysis at the end. So we expect that this might happen once every year or 2 years. But for now, I think there's a greater urgency to do it annually, at least for the immediate needs as such.
Yeah, maybe just to add one point, I think we're really dependent on you also to flag unusual things to us, because one of the difficult things about tuberculosis is that it's not well mixed. You can have a clone that is very frequent in one setting that you don't see anywhere else. So the sensitivity and specificity that we're calculating in this catalog are specific to the collection that we have. In your setting, it might be completely different. So a good example would be the 491 RBOB mutation, which is extremely frequent in Eswatini, but very rare elsewhere. So the performance characteristic of, say, GeneXpert is very good in most settings, but very poor in that particular setting. So I think it's going to be, as we go forward, we'll spend a lot of our effort to chase more unusual things, which are unusual globally, but can be very prevalent locally. And we've already tried to do that, for example, by including the GIRA-A90G mutation, which we know causes a problem in Central Africa, to really respond to needs in the field. So we really do need you to flag unusual things to us. And this is why I think, because you ultimately are the experts as to what is going on within your individual countries. And if you find unusually poor performance of a particular assay, you find something very unusual or a strain that appears to be increasing in frequency relatively rapidly, then please do reach out to your reference laboratory or to us so that this can be followed up and dealt with. So ideally, we want to transition from a scenario where we go out to collect things to a sort of genome-driven scenario of microbiology where we respond to needs as they arise, because ultimately the best signal for clinical relevance is increasing frequency of a particular strain. Much like for COVID-19, the response is driven by typing and sequencing currently. So we want to move to a sort of real-time scenario ultimately, ideally.
I'm going to jump in here. I see there's a question there from Yves. Some mutations are missing in the mutation catalog, but we can see the mutation name in the LEGENT catalog. What can we do? Okay, so there we go. A specific example. Paolo, you want to jump in and play with that on your screen and show what's going on?
So the mutation is the one of the pole. So to look for synonymous mutation, you have to switch to the genome indices here. And as I said before, synonymous mutation were considered wild type, and so that were not graded. And if you look here for your mutation, that is GBL103L, you can find it here. So you have anyway the mutation reported in the genome indices spreadsheet, but since it was not among the mutations considered for grading, it is not reported in the mutation catalog. So in this spreadsheet, synonymous mutations are not reported, but are reported only here for, let's say, completeness of the information. I hope this answer Yves' question.
Yves, let us know if that if you were able to get that to work.
Okay.
I'll take that as a yes. I see there's another question up there that's going to be very difficult to answer. Claudio, could you tackle that question about, do we still need phenotypic testing?
I think the answer to that, it depends. So I think for mutations that we understand well, don't think there's not much point in gathering more data, especially if you need to spend extra money to do it. So if it's part, say, of a plate and you have to include it because you're interested in something else, then that's one thing. But I think in some cases, doing phenotypic testing can be counterproductive, namely for mutations that confer borderline phenotypes where the phenotypic testing is not a reliable confirmatory test. Because if you have an isolate that comes out as resistant, say with one of the 7 borderline resistance mutation for vancomycin, where WHO has now explicitly said that phenotype is not a reliable confirmatory test. So not only can you, so phenotypic testing can be counterproductive in those circumstances, but I think especially for things like bedaquiline, but also the nitromidazoles, I think we're going to have to keep on doing phenotypic testing for novel mutations that we find to build up the level of evidence surrounding their association. The good thing is that in most settings, at least MDR clones are highly clonal. So even though there might be lots of mutations globally, in an individual setting, the spectrum is somewhat limited. So once you've seen a dominant clone and have gotten a couple of phenotypes, you can sort of safely classify that mutation. But it really also depends on your workflow. So we can certainly reduce phenotype as much as possible, especially given if the initial test doesn't give any indication of resistance. But I think for high value patients, MDR, that have unusual mutations, we do need capacity to do it. So it'll be a triage approach as to where it is needed, I think.
Thank you, Claudio. I completely agree. That's a depends question. Paolo, could you answer the question about what about mutations in bovis genome? Can you tell us how bovis was handled in this?
So this catalog is including all the species belonging the tuberculosis complex. So there are also bovis mutations, and one example is the PNCH57D, that is a marker for the bovis. So it is considered within the complex, is not MTB and tuberculosis specific, let's say.
Can you pull it out by species? Is there a way, I can't remember in the Excel or anything, to pull it out by species?
No, this is not tracked in the catalog.
And then referring to the specific question here, we have detected mutations in bovis genomes. How could we compare them? What would your answer to that be?
This is a nice question. I think that the only way is if the numbering is different, you have to translate the M. bovis numbering to the tuberculosis numbering, if in case the gene you are looking at is different. Otherwise, this is an M. tuberculosis complex catalog. So if the mutation is the same, happening at the same number, you should be able to find it look at it.
So they have an embos mutation. Just make sure that they're using the right numbering, the same numbering system as the catalog, and boom, they can compare it and look for it. So I hope that answers your question. Please let us know if it doesn't. Okay, additional question on pyrazinamide DST. Neither genotypic nor phenotypic is very reliable. What can we do in these circumstances? Claudio, this is an excellent Claudio phenotype genotype.
Again, it's both phenotypic and genotypic testing for pyrazinamide have their strengths and weaknesses. So the catalog provides clear guidance as to how to interpret the mutation if you find them up front. There are some mutations that confer borderline phenotypes, which are therefore difficult to confirm. And I think this is something where we need to provide more guidance. I think the other point for pyrazinamide is that actually the current breakpoint that we use might actually be too low because lineage 1 strains appear to have intrinsically elevated MICs, and therefore what our sensitivity may actually be much higher than it actually is as currently calculated. So again, I think there is a need for us to provide better guidance as to whether phenotypic testing is needed at all, depending on what we find up front, especially given that testing pyrazinamide is a conscious decision. It's, you need to set up the pyrazinamide kit to do it. And so we need to provide guidance as to whether it's needed, how you deal with discordances, and borderline mutations are a problem. In fact, and if you read the pyrazinamide section, we had actually misclassified one mutation as not being associated with resistance in the sequencing guide because it had sort of a borderline character, but we've now corrected that. So again, I think it needs more work, but I think the sensitivity of sequencing is actually rather good. But it does need more guidance. I think there was a second question regarding borderline resistance mutation don't always test resistant even at the lower MGIT breakpoint 0.5 milligrams per liter, and that's correct. And that's basically because the breakpoint, so it depends slightly on which borderline mutation you have. There are some that are more likely, that have higher likelihood of testing resistance at the new critical consideration, others less so. But they all confer elevated MICs. But none of them can be confirmed reliably because the MIC distribution is divided by the breakpoint. So because of the inherent variation in testing, so for some mutations, you might as well toss a coin as to whether it comes out as resistant as susceptible because the distribution is divided right smack in the middle by the breakpoint. And here WHO guidance is crystal clear. If you find one of these mutations, it is resistant, you can't use RIF, it needs an MDR regimen. This will have to be reconsidered should a higher dose of RIF be endorsed, but that's not currently the case. So in this particular case, you can also read at the last section of the systematic review of the critical concentrations for rifamycin and isoniazid as to the justification. That rule has already been included in the mutation catalog. And there's a detailed justification as to why the committee took this particular decision. And this is also why because if you remember in the literature, they're often called disputed mutation because there's always some sort of doubt as to whether they cause resistance or not. So the committee felt that they do confer resistance, but because of the borderline nature, they can't be confirmed reliably. That's why they were renamed, but they definitely do confer resistance.
Great. Thank you, Claudio. We're coming up on the half hour, Nazir. Do you want to keep the session open a little bit longer, or should we officially close? One more question.
I think we can take the last question, and I think we should round up. It's been like 3.5 hours.
I think that sounds like a good idea. Right. Okay, let's take this last question. Can we confirm RIF status by phenotypic DST by liquid culture? Claudia, I'm going to give that one to you again.
So, I mean, it really depends what you say by confirm, right? If you have a mutation like the S450L, that confers large MIC increases, more than 100-fold MIC increases. So that is a mutation that, unless you make a mistake setting up your phenotypic testing, or you've done a mistake using your upfront genotypic test, then of course you can confirm that. Whether that is a good way to use limited resources is a separate question. So high-level resistant mutations are easily confirmed. Borderline mutations, phenotypic testing isn't a reliable confirmatory test. And then, and I think this is where I think we need to be a little clearer as to discordant between line propagating gene X. But okay, so that's another issue, because obviously you can get systematic false resistant results by synonymous mutations, because most synonymous mutation in RPOB, in the rifampicin resistant Z-type region, confers false resistance. So the best way to exclude that possibility would be to use sequencing, because if you only find a synonymous mutation, then you can be sure that your resistant result was for that reason. And this is also why WHO now differentiates between resistance inferred, in other words, where you only infer resistance by lack of binding of the probe, versus if you have actual mutation probe that binds, where you can be sure which precise mutation is found. So it really depends as to what you are trying to deal with. And this is why I think targeted Nextgen is such a useful reflex test for anything that looks to be resistant based on your decentralized testing, be it Gene Expert, the Ultra, or the XDR cartridge. You could then, once endorsed, reflex it on sequencing. You'd immediately be able to spot cases with synonymous mutations. And these are rare in most settings, but not always. For instance, in Brazil, they actually have 2 clones with 2 different synonymous mutations that are very frequent, which means that the positive predictive value of these rapid tests is poor. So again, that's why sequencing gives you so much more information as to what you are dealing with.
But for the vast majority of the world, getting an expert or an LPA with a rifampin resistance based on mutation, you can take that to the bank. Yes or no?
Yes, taking into consider the positive predictive value, and WHO has guidance about, because it depends on the pretest probability of your patient. So whether you're in a high, whether he or she is likely to have MDR, TB or not. So you wouldn't always act on a single expert result. You need to take the pretest probability into consideration, but you can look that up in the diagnostic manual.
Operational handbook.
Great. And with that, I think everyone's been on for a very long time. Thank you very much, Claudia and Paolo, for staying on extra time to take people through some questions and last minute questions and issues around the catalog. Thanks, Nazir. I think that's it.
Yeah, I think it's been a long. I'd just really like to thank all the people that have contributed to the catalog and spending time with the group today. So it's been a fantastic session. Thank you very much. Have a great rest of the day. Bye.
Thanks, everyone.
Thank you very much.
Thank you. Bye.
Thanks, Caroline. Bye.
Bye.